Good morning, everybody. Great to see a crowd raring to go this morning for a great morning. The 2024 Canada Gairdner Award laureates are going to be talking about their prize-winning research. We're also going to have, scattered amongst the laureates, 3 early career investigators across Canada who've been selected by our awardees and will give short talks on their research as well. So we have the next generation represented. We also have the next generation represented in the front rows here with a number of high school students, so welcome to you as well. Hope you enjoy the day. It's my pleasure to welcome you in person. It's great to see so many people. We also stream this, so I know there are people watching online, so hello to you too. And before we begin, I'd like to take a moment to acknowledge that MaRS is located in Toronto on the traditional territory of many nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat peoples, and is now home to many diverse First Nations, Inuit, and Métis peoples. This territory is subject to the Dish of One Spoon Wampum Belt Covenant, an agreement to peaceably share and care for the Great Lakes region, and we are honored to share the land today. This event is part of Gairdner Science Week, an annual week-long celebration of our Gairdner Award laureates and the impact of their research on human health. I'd like to thank our longtime Gairdner partners, the Government of Canada through CIHR, the Government of Quebec, and the Government of Alberta. And a special thanks to all our Gairdner Week sponsors who've made this year's event possible. They're listed for you as you walk by. They are our platinum sponsors, the Kremble Foundation, the Institute of Philanthropy, and TELUS Health. Our gold sponsors are the University Health Network, the University of Toronto, Women's College Hospital, London Drugs, McMaster University, the International Development Research Centre, Sunnybrook Research Institute, Moderna, and the Centre for Addiction and Mental Health. Thank you, and thank all of our sponsors and all the volunteers who have been working across Canada, welcoming our laureates, who have all been working very hard for us this week. And maybe they'll get a chance to relax because we have the gala tonight. But this morning, we're going to make them work again. So you get an award, but you have to come and work for the Gairdner Foundation. I'm truly thrilled to welcome 7 of our 8 2023 Canada Gairdner Award laureates in person to join us today. Unfortunately, Dr. Eshar, who shares the award with Dr. Michelle Sutherland for CAR T cells, is not well enough to be able to join us today, won't be able to present. And as I say, they've been across the country, and they're here now in Toronto. Now, without further ado, let's hear about the prize-winning research of our laureates. Please feel free to follow us on X and Instagram at Gairdner Awards. Our first 3 lectures are going to be given by the laureates who are being recognized for the development of so-called next-generation DNA sequencing technology. So Shankar Balasubramanian is the Herschel Smith Professor of Medicinal Chemistry at the University of Cambridge and Senior Group Leader at Cancer Research UK's Cambridge Institute. He's co-inventor of the leading next-generation DNA sequencing technology, Solexa sequencing, now Illumina, that's made routine, accurate, low-cost sequencing of human genomes a reality and has revolutionized biology and, as we heard yesterday, is revolutionizing medicine. He's invented chemistry to decode several modified epigenetic DNA bases and DNA secondary structures in the genome, and he's made seminal contributions towards the understanding of their dynamics and function. His work on small molecule recognition of nucleic acids has revealed molecular mechanisms that can be exploited to modulate the biology of cancer. His collective contributions span fundamental chemistry and its application to biological and medical sciences. Sir Shankar was knighted in the Queen's New Year's Honour in 2017 and was awarded the Royal Society's Royal Medal in 2018. And in 2023, he was elected as an international member of the National Academy of Sciences, USA. So our 2024 Canada Gairdner International Award laureate, Shankar Balasubramanian, will be presenting Reading Your DNA. Please join me in welcoming him to the stage. Shankar.
Not a very good start. Thank you, Janet. Honored to be here and be a recipient of the Gairdner Prize, and thanks to the foundation. Can you hear me? Yeah, good. Okay, I'm going to give you my perspective on some of the challenges that we needed to overcome to develop the sequencing. I should say my part of the work was a collaboration with David Klenneman, who you'll hear from next, and it took place in the Department of Chemistry and led to the foundation of a company called Solexa, where the technology was developed into a fully integrated commercial system, and then Illumina acquired it and developed it further. These are actually other companies that have spawned out of very basic science projects in my lab. Now, Cambridge is actually famous for this structure, the DNA double helix, which in 1953, there were two important papers published. Watson and Crick did the interpretation, and Franklin and Wilkins generated the X-ray data. And the molecular basis for the genetic code boils down to this very simple relationship of the C base recognizing the G base through hydrogen bonding and the T base recognizing the A base. And it's remarkable, the whole of genetics boils down to this very simple molecular relationship. And DNA, of course, is a string of 4 letters arranged in a particular sequence. One copy of the human genome is made up of 3.2 billion of these letters organized in 23 chromosomes. And units of these letters that make up genes, these are typically thousands of bases. The genes encode for proteins that are the machines and hardware of cells. And that forms about 2% of the genome. The other 98% used to be dismissed as junk, but we now know that it's important in orchestrating and regulating how genes are activated. Now, the question here was the basis of the Human Genome Project. Why sequence DNA? Well, this blueprint provides the foundation for understanding living systems. Now, all living systems have a genome. That's the entirety of the DNA. Viruses, some viruses have an RNA genome. So sequencing it provides a foundation of knowledge. Now, one of the drivers for the Human Genome Project was to understand the genetic basis of who we are and why we're different, and why some of us are predisposed to diseases, and of course some diseases are caused by genetic changes to our genome. Now, many years ago, David and I paid a visit to the Wellcome Trust Sanger Institute, which is where the UK part of the Human Genome Project was taking place. Now, we had questions. At this stage, they were about a quarter of the way through their part of the project. But one of the questions was, to understand these things, you need to sequence at a population scale, not just one reference genome. Now, at this point, this was in the 1990s, it wasn't clear quite how that would happen. So we shared with them some of our thinking. Now, our start into this project has nothing to do with DNA sequencing. We were carrying out a rather abstract, pure, fundamental experiment. So you'll see David, and he'll explain the riddle of why he's holding a pint of ale in this photograph. And our postdocs, Colin Barnes and Mark Osborne, were key. Now, I'm a chemist, but along the way, I had gotten interested in understanding the chemical details of how enzymes catalyze reactions. And during my postdoc, I got interested in these wonderful machines called DNA polymerases that copy DNA every time a cell divides. And one of the beautiful properties of DNA is it can self-replicate, it can template its own synthesis. So we dreamt up an experiment. Now, David is a physical chemist, as you'll see in his talk, and single molecule fluorescence detection at room temperature was a relatively new observation technique at that point in time. So we wrote a grant to this research council saying we want to use this new observation technique to watch nature's machines make DNA and see what we can learn from that. That was our only aspiration. There was nothing useful implied in what we were proposing to do. We were just trying to see what we could learn. Now, the setup, they funded it remarkably, and I'm not sure they would fund that same grant today. Now, the setup included a glass surface, and we immobilized synthetic DNA. Here's the template, here's the primer. This oval is the machine, the polymerase, and we watched this strand being extended one building block at a time. And the way we did this was by using fluorescently labeled building blocks. And the fluorophore was the observation method. Now, after our talk with the people at the Sanger and thinking more about what we were doing, we saw that what we were doing actually could be used to read the template strand of DNA by color coding the building blocks. So this is the concept behind what we did at Solexa. This same setup, here's the template, here's the primer. If we could persuade the polymerase to incorporate the correct base, now here T is the next templating base, here's where the building block goes. So you would bring this red dye with it. Now, if you could control this reaction so it would stop and persuade the polymerase to do it, you could then image this surface and you'd read off a red dye, which would give you the identity of the first building block that's gone in. Now, if we could remove this dye and remove what was preventing the second building block from going in, we would be back here in the n+1 position, and in the next cycle, we could incorporate a T opposite an A, bring in this dye here, and read a second color. So cycle by cycle, you would see a series of changing colors at this single molecule. And in doing so, you were actually decoding the sequence of the building blocks in this template. Now, that's reading a piece of DNA that's attached to a surface. The way we were doing these experiments involved imaging lots of DNA molecules on a surface. Now, our vision for turning this into high throughput sequencing was extract DNA from a cell, fragment it into small pieces. Now, by controlling salt and dilution conditions, you can monodisperse these fragments on a surface so that each molecule of DNA would hit the surface and attach and stand in its own optically resolvable space. Now, you could then sequence all of these in parallel at the same time and retrieve a lot of information by scaling. So this was the principle that led to scaling DNA sequencing. Now, this was a crude calculation. It turned out to be dimensionally not quite correct, but the spirit of it was correct. So a human genome is of the order of billions of bases. That was the challenge. So if we assumed an array, and we assumed a rather modest array, we had a good understanding of the kinetics of some of these reaction steps. So we assumed a cycle time and calculated that you could sequence of the order of a genome on a time scale of days. Okay, so this was our prediction, billion base sequencer. Now, I'm going to tell you the essence of how this works and was reduced to practice in Solexa. So here's the primer, here's the template. The way a DNA polymerase works is it extends off this oxygen to incorporate the next building block. And I'm just going to show a little bit of chemistry here because I know there are some chemistry students at the front who might find this interesting. So this oxygen here acts as a nucleophile. This is a monomer. I've just put base. This depends on whether it's a G or C or T or A. And this oxygen actually attacks this phosphorus, and this diphosphate is a leaving group. And the enzyme, which I've simplified here, holds two magnesium ions in place, and they are actually the catalysts for this reaction. And you add one building block. So we had to hijack this process, and there were two aspects to this. One was re-engineering the building block into something unnatural. And the other, which I won't go into details about, was re-engineering the enzyme, the polymerase, to tolerate these unnatural chemical features in the building blocks. Now, there were two things that needed doing to the building block. One, this oxygen, which is the nucleophile for the reaction, we had to protect it on the monomer so that it would incorporate but not allow a subsequent incorporation, because we need to stop and acquire the information by reading the fluorescence. So we put what chemists call a protecting group on here. Now the fluorophore we attach to the base at the edge that is not involved in Watson-Crick hydrogen bonding. So on C and T, we attach at carbon number 5. With G and A, we do what's called an N7 diazo replacement, and we attach there. Now, the chemistry here needed to be water compatible and mild, and a reaction that does not cross-react with DNA. And this was actually a lot of hard work went into this because it's a non-trivial problem. And it needs to be really efficient if you want to sequence lots of bases of DNA. Now, the chemistry of the first generation sequencing nucleotides developed at Solexa used this group here. This is an azide. Some of you will know of AZT, which was the first nucleoside drug against HIV-1. Now, AZT has an azide attached to the ribose. This is an azide and then a carbon and then an oxygen. And there's a reaction invented in the early 1900s by a chemist called Staudinger, where he showed that phosphines, which are these reagents, can reduce an azide to a primary amine. So an azide is a disguised primary amine. If you reduce this, this is called a hemiaminal, and in water it hydrolyzes spontaneously, like so. So we can cleave here, and we can cleave here, releasing the fluorophore with this reagent, the phosphine. Now, this is our intersection with what Pascal was doing at Mantea, and Pascal will say more of this. There was an important change to our original vision, which was single molecule sequencing. And in fact, we adopted this lovely method that Pascal and his colleagues developed. I won't go through the detail, but in essence, it takes a single molecule attached to a surface and then amplifies it into hundreds of copies of identical sequence. But they all stay in the same spatial location. This is hugely important. Now, the reason we looked to amplify fragments was single molecule detection is beautiful because it allows you to observe stochasticity. Now, stochasticity, unfortunately, was leading to errors in reading these sequences. And I'll mention his name because he's a former Gairdner Laureate. The late, great Sidney Brenner, who often spent time in Cambridge, he once, he called me to his home in Ely, and I had a conversation with him in his kitchen of his house, in which he very forcibly said we should be amplifying these molecules. And in fact, he suggested a way, but really the best way of doing this is this method shown here. So this was adopted into the final sequencing system. Now, 2005 was around the time we did the first genome sequencing. It was a small genome, Phi-X174, which is a bacteriophage. And shown here is an expanded image taken by a charge-coupled detector imaging the surface at one cycle. And these round images here are actually amplified clusters of a single molecule of DNA. And the colors tell you what the base identity is for that step in the sequencing. So cycle by cycle, each of these spots would change color according to the next base that's been incorporated. And you can image, transfer, convert, and digitize the information into sequence. And you get strings of sequences from each site on the surface. Now, back in the 90s, when we were trying to look forwards, one of the questions was, how long a piece of DNA could we read? Now, we did some calculations that suggested if we could read at least 25 bases, not knowing where the fragment came from, even if there were one or two single nucleotide polymorphisms, given the Human Genome Project was going to provide a reference sequence, we could computationally realign this to the reference to find out where it came from and cover most of the genome. So the challenge became to get at least 25 cycles. And you can see here the fragments get realigned and you can then read off the sequence. Now, all of that system today works sufficiently well to read hundreds of bases, and typically you read 250 from one end and 250 from the other end. This is called a paired read. Or today, I think it's possible to read over 500 bases in a single read. I think 600 is the latest system. Now, you can reassemble a genome. In 2008, there were 3 genomes published in one edition. One which we were involved with was the first African genome, Yoruba genome. There was also the first Asian genome, and there was the first cancer genome. And they were all done at that time, reading 35 bases from one end and 35 from the other. It was possible to construct a genome. Now, this doubled the number of genomes deposited in the database in just one edition. In 2006 was when Solexa released the first system. It was called a 1G genome analyzer. It looked like this, and you'll find these in museums now. They're already in museums, they've been relegated. But they sequenced 1 billion bases of DNA in a single run, which at that time took a few weeks, actually. It was not so fast. But it achieved the 1 billion base prediction or promise. Now, these higher-end instruments that were introduced by Illumina in 2021 had further optimized, engineered, improved the process at every level. And these beasts sequence trillions of bases of DNA per experiment. Overall, this is it's actually about a 10 million-fold improvement from the mid-90s when we were first trying to visualize this whole program of research. So over a million-fold improvement means sequencing the equivalent of about a genome a day, a genome an hour on these instruments, actually. You can sequence tens of genomes in 24 hours. And the vision of population scale sequencing is actually here. There are programs in many countries around the world where sequencing is being done at scale. This is one, the International Cancer Genome Consortium in the UK. Genomics England is a branch of our National Health Service, state-funded healthcare system. Other countries, Finland, the US, and of course Canada as well, have large-scale clinical sequencing programs. Now, I think David's going to say a bit more about application, but I'm just going to give you a taste in the cancer area. Now, this is a sort of a textbook example of a very specific mutation in a gene, BRAF. And this mutation causes the gene product to become overactive and drive cellular proliferation without control. And a certain proportion of people who have melanomas, skin cancers, have this very specific mutation. And there is a drug that acts on the protein and inhibits it. So patients that have this mutation respond very well to that specific drug. So this is an example of if you know the genetic characteristics of the tumor, you might be able to treat that tumor in a very specific and personalized way. These projects have generated and are generating enormous amounts of data linked to patient records. And what's emerging for some tumors is patterns of genetic changes that characterize the tumor and allow physicians to make an informed decision about treatment. But it's an ongoing project, and I think over the next 10 to 20 years we will see the full extent to which such information can help us deal with diseases such as the many types of cancers. I want to say something about early detection. Most diseases, and in particular cancers, it's desirable to catch it very early, and many of them are treatable with good outcomes if you can catch them early. And a burgeoning field is actually based on the remarkable observation that in blood there is cell-free DNA in the form of fragments circulating that can be retrieved from a blood draw, sequenced at great depth. And in patients that have tumor, the tumor cells necrose and release their contents into the plasma. And floating around are DNA fragments that originated from the tumor that can carry characteristic changes to the DNA, which may be either mutations or actually more commonly now, people look at what are called epigenetic characteristics of the DNA. And there are ongoing clinical trials now looking at patients and actually members of the population who don't show any signs of disease. And there are many cases of diagnosing disease. The holy grail here is to actually catch cancers at early stage, at stage 1. We still have some work to do, but this is an area to watch out for. This is my last slide. I'm just going to say something about what else I do in my, I was going to say in my work, but actually science is not work. It's a hobby and an obsession, and this is what else I've been doing. I've spent the last 30 years working on DNA, and I'm a chemist, so I'm always interested in the molecular aspects of DNA. And in that very first slide, I said to you, DNA is molecular information. It turns out that the order of GCAT in DNA is only one form of information that DNA stores and can convey. I'm going to give you two examples of areas that we work on. One is on structures. Now, I showed you the double helix structure of DNA. It turns out that is not the only structure that DNA can assume. And there is a quadruple helix structure shown here called a G quadruplex. And we've spent quite a bit of time elucidating these structures. They form in human cells. They tend to be very abundant in cancer states, actually, and they are indicative of cell states. And we recently provided evidence that they are part of the process that control the activation and inactivation of genes. So this is one area that we work on. The other is there's some interesting covalent chemistry that occurs naturally in DNA. You know, I alluded to a term epigenetics. Well, this is one of the four bases, the C base, and there is an enzyme that can put a methyl group here to form this methylated cytosine derivative, 5-MC. And this can actually be oxidized by another enzyme to this alcohol here. These modified bases exist in your DNA, in every cell in your body. And part of what these things do is, again, they are involved in orchestrating whether genes are switched on and switched off. And it's a grayscale, it's quite complex. And they help set the identity of cells. And this is part of why cells in your skin differ from cells in your kidney, cells in your heart, and so forth. They all share the same genome, but the epigenome reprograms them differently. And in the onset of disease states, you get changes in these epigenetic bases. So part of what I've been working on here is chemistry to sequence these modified bases. And I have to disclaim here a company called Biomedical that I'm involved with. They now have a sequencing technology that I helped develop that gives you a 6-letter human genome, where you get GCAT, MC, and HMC. And I predict, particularly in early onset of disease, that this is going to be quite important. So I'm going to stop there and give some thank yous. Firstly, the team in the university, superb team in Solexa, and also Illumina, who improved the technology further. Lots of people contributed to what I've just described, and I'm representing all of them, so credit to all the players. This all started with basic science. We weren't actually trying to do anything useful. And I think it's very important, especially for the young people here, to not be afraid of pursuing science for its own sake and ask curiosity-driven questions. You never know where it will lead you. I think it's also important for the politicians and funding agencies to acknowledge. I've already mentioned the team, and lastly, the technology has been used and applied by millions of people around the world, including those involved in clinical research, clinicians. Patients have given their DNA for some of this work. So really, the impact and outcomes of this is a major team effort that hopefully will help solve problems. Thank you very much.
Thank you very much. No questions.
No questions. Okay.
No questions, sorry. We're not doing questions this morning. You can grab the laureates at the break. I'd now like to introduce our next laureate, Sir David Klenerman. He's the Royal Society GSK Professor of Molecular Medicine at the Youssef Hamid Department of Chemistry at the University of Cambridge. A physical chemist, he completed his doctorate at Cambridge University with Professor Ian Smith on infrared chemiluminescence in 1985. Did postdoc work on high overtone chemistry with Professor Dick Zare at Stanford. And after 7 years with BP Research in their UK laser spectroscopy group, he returned to the University of Cambridge, and there his work was focused on the development and application of physical methods, particularly laser spectroscopy and single molecule fluorescence, as we're beginning to hear, to biological and biomedical problems. Recently, he's focused his work on studying the molecular basis of the adaptive immune response and detection and characterization of protein aggregates associated with neurodegenerative disease. Dr. Klenerman was knighted in 2018 for the development of high-speed DNA sequencing and also awarded the Royal Society's Royal Medal in 2018. Well, let me ask you all to welcome Dr. David Klenerman to the stage and present on single molecule fluorescence and its biomedical application. David.
Thank you very much. It's a real pleasure to be here today. So what I'm going to tell you is a little bit about my background, and then I'm going to explain to you a little bit more about single molecule fluorescence that we've already heard about this morning. Then I'm going to talk about DNA sequencing, largely focusing on its application, since you already heard from Shankar the key ideas behind the technology. And then in the time left to me, I'm going to talk about how we're also using the same methods to study protein aggregation and neurodegeneration. And then one of the key themes I want to emphasize in the talk is how physical chemists build new instruments that enable us to measure new things and how that can enable new science. So this is me when I got my PhD, my undergraduate degree. So I wanted to study chemistry. I found biology too qualitative at the time and physics too abstract, so I focused on chemistry. And then I did a PhD. So this is the instrument that I built. So there's all this gas handling that I made myself. There's a home-built infrared spectrometer, and there's this big reaction chamber here. And what we were studying was hydrogen atoms that we made by microwave discharge, interacting with ozone, and that produced OH radicals that were vibrationally excited, and we used the spectrometer to detect the infrared radiation that these molecules emitted. I then went to the States, where I worked with Dick Zare. So what I learned from Dick Zare, he had an infectious enthusiasm for doing science. He was like a kid in a candy store. He really just loved doing science. And he really wanted to empower his students to do the best science they could. And that's something I've tried to do in my group. So in Dick's group, I tried to selectively cleave bonds in an organic molecule using a laser, which wasn't that successful. It's a very good way of heating the molecule up, but not cleaving the bonds where you wanted them. And then I returned to BP. And so at BP, I learned how to do multidisciplinary research. I was a laser spectroscopist, but I worked on very applied problems like how do corrosion inhibitors work, which the company used in the Alaskan pipeline to stop them corroding. So what I did here is I had this large laser table. If you've never seen a laser lab, we're normally in the basement in the dark. Here's a laser, a green laser. There's a dye laser here that's tunable to change the wavelengths. This laser here amplified the power of these pulses. Then it's relayed down to the sample, which in this case was a piece of iron in salt water, which was being corroded. And we were trying to watch the corrosion inhibitors bind to the surface of the corroding metal and see how they work. And then after that, I returned to Cambridge in the mid-90s. And at the time, one of the most exciting things happening in chemistry, and certainly in physical chemistry, was the capability to see single molecules. And the way that was done is as follows. You have a laser pointer exactly like this. You can even use a laser pointer. And we have a dye molecule, which is fluorescent. So if I excite the dye molecule, it will fluoresce a yellow photon. And I can do this process again and again and again. So typically, a single fluorophore can produce 1 million to 10 million photons, which are easily detectable with modern imaging technology. So a single fluorophore produces 10 million photons, I can easily detect it. And if I attach my fluorophore to a molecule of interest, a nucleotide, for example, or something else, then I can detect that molecule. So you work at very low concentrations, typically picomolars, so only one molecule is present at a time. And as Shankar showed you, it's the ultimate in miniaturization, and that's the idea behind this massively parallel way of sequencing DNA. But at the time, we weren't interested in sequencing DNA. Like Shankar's told you, we were interested in watching biology in action. So this is, again, another laser lab. It's all very colorful, purple, Cambridge blue, green, and red lasers relayed over to an inverted microscope. The microscope has an objective, and you might have learned in your physics class that light at a certain angle undergoes total internal reflection. But a small amount of the light, there's a decaying field, an evanescent field that propagates a couple of 100 nanometers above the glass surface. So here we have a T cell, which you're going to hear more about later on in the symposium. And this is a live T cell, and we've got an antibody that binds to the T cell receptor, which is responsible for initiating the adaptive immune response. And the antibody is labeled with a fluorophore. And so what we can do is watch the T cell receptor diffusing around on the surface of this live T cell. So this is what it's going to show you here. So each of those spots is a single molecule, and by analyzing this data, we can ask how do the T cell receptors diffuse around on the cell surface, or they're monomeric and dimeric, and how do they reorganize when in the initial early events of the adaptive immune response. So like Shankar's told you, we came up with this idea of using this technology to do a very fundamental study of how DNA polymerase incorporates nucleotides as it copies DNA, using two postdocs, Colin Barnes and Mark Osborne. And at the time, like Shankar's told you, it cost $3 billion to sequence the first consensus human genome, and then $30 to $50 million to sequence in individuals. And there was a real need, interest in getting this down to around $1,000 a genome. So we went to the pub. There's a local pub, the Panton Arms, just very close to the chemistry department. And the pub is not the important bit. The important bit is we talked to each other and I learned more about DNA and DNA polymerases. Shankar learned more about single molecule fluorescence. And that's how we came up with the idea of Celexa sequencing. The alcohol maybe have helped a little bit, but I think it's mainly the talking to each other. So we came up. So we didn't know quite what to do when we came up with the idea, whether we should write research grants. But one of my colleagues, who was a master of Christ's College at the time, Alan Munro, persuaded us that we were going to need increasing amounts of money to develop the technology. And hence, we decided that we were going to form a company, and the company was called Celexa. So it's a play on words. We had single and mol, and Shankar thought any good company had to have X in it, like Xerox. So we came up with the idea of Celexa. And we were thinking of having these DNA hairpins, so this is where this structure comes from. So, and it turned out that Alan gave us very good advice. So we came up with the idea, like Shankar's shown you, in 1997. In the year 2000, that there's this fly talking to some lizard at a garden party saying, well, have you had your genome sequenced? So this was just when the first average sequence of the human genome was coming out. In 2009, like Shankar's told you, we published our first human genome of an individual published, which was generated using our technology. So that's very rapid 12 years going from an idea literally on a piece of paper to something that works and is published in Nature. And we couldn't have done that without forming the company. So it's gone off, the speed of uptake is well beyond what we imagined when we first came up with the idea. So this is a plot of number of genome sequence. It's a log scale against time. So 2001, the first genome. In 2007, when we published our genome, you can see that it really takes off. And it's now gone up to about 30 million genomes that have been sequenced, about 90% using this Alexa technology. Similarly, the cost has come crashing down, so up from millions of dollars all the way down now to $200 per genome. Like Shankar mentioned, in the UK, there's a 100K genome where they're sequencing individuals and then following their health records over time to understand the genetic contribution to common disease. And probably the most striking thing for me is this study. It's being used for newborn babies. So this was a newborn baby born in Addenbrooke's. So it's the baby, the mother, and the father. And this baby was unwell when it was born, but it was sequenced using the sequencing technology. They sequenced the baby, the mother, and the father. And they can identify a gene that had been mutated. And in this instance, there was a treatment for the baby. And so on the television, they showed a very healthy baby and two very happy parents because they managed to cure the baby. And this has been commonly used, certainly in the UK. In about 50% of the cases, they can identify what's wrong with the baby and they can be treated. The technology was used widely in the pandemic to look at how the COVID virus mutated and to then start to design new vaccines. So this is a plot of time, 2020 to 2021. Again, a log scale of the number of COVID virus sequences that were taken, again, largely using this Alexa technology. And I just wanted to highlight another interesting application. So this is Svante Pääbo, who won the Nobel Prize in 2022. And he developed a way of taking very old DNA from bone samples. So here's the Neanderthal, and he took a bone sample and managed to extract the DNA and sequence it. And again, he also took a sample from a cave in southern Asia. And he was being able to identify, sequence the Neanderthal DNA or the Denisovan DNA. And from that, we could learn about our ancestry. So we had a common ancestry, and then we separated. This is Homo sapiens, this is Neanderthal, this is Denisovans. And so what he found is about 2 to 4% of our genome comes from our Neanderthal ancestry. So that's just another use of the technology, learning about where we came from. So in the other part of my talk, I want to talk about what we're doing now with the same technology. So we're very interested in understanding the molecular basis of neurodegenerative disease. So in neurodegenerative disease, you get these extracellular plaques, these intracellular tau tangles, or you get these intracellular Lewy bodies made up of alpha-synuclein. But it's not these sort of microscopic aggregates that cause the disease. It's the much smaller aggregates that have to form on the way to forming these larger aggregates that are thought to be toxic. And so we've developed methods to characterize these toxic aggregates, which are present at very low concentrations. And so we really need the single molecule sensitivity to do this. So this is not a cartoon. This is actually from a paper we published in Cell, where we aggregated alpha-synuclein in the test tube and worked out the mechanism. So I'll just talk you through this briefly. We start with monomer here. It forms small dimers and trimers by addition of monomer. And then what we found, these so-called oligomers can undergo a structural conversion into a more fibrillar-like aggregate that then can grow by addition of monomer into the large fibrils that make up the Lewy bodies. And what we also showed is that these small fibrillar-like aggregates were much more toxic than these aggregates. And this structural conversion was actually very slow. It took 24 hours, giving the cell some time to get rid of these aggregates before they converted into more toxic form. So, and then we had a sort of eureka moment in our study of protein aggregation, which was the experiment one of my students, Matthew Horrock, did. So what we did is take cerebral spinal fluid. So this is the fluid around a person's brain that you get from a lumbar puncture. And so we took the cerebral spinal fluid of a patient unfortunate enough to have Parkinson's disease and put it down on one of our sensitive microscopes. And so all we did was dilute it tenfold and add a dye, thioflavin T. So thioflavin T is non-planar in water and non-fluorescent, but it becomes planar and fluorescent because it can intercalate inside the aggregates, inside the beta sheet, and so it becomes highly fluorescent. And when we did this experiment, we saw these small spots, and each of these spots we think is a beta sheet containing aggregate. So we were very excited because now we could look at the real aggregates that form in humans, but we couldn't measure their size because they were blurred by diffraction, and we couldn't learn about their composition because thioflavin T binds to any aggregate that forms a beta sheet structure. So I just need to have a slight detour to explain to you the other advance in my field, which is how you can break the diffraction limit and do super resolution microscopy. So this is an image blurred by diffraction, but what I can do is organize that I just have a few molecules fluorescing at one time. Again, this individual molecule is blurred by diffraction, so it's a 200 nanometer spot. But I know it's a single molecule, so I can replace this blurry spot with this higher precision spot. So typically, I can localize the molecule to 20 nanometer precision. And then if I do this again and again and again, I can build up a super resolution picture of the object of interest. So we're going to use that to look at our aggregates. So this is how we did it. We have a pegylated surface, so this is essentially a very non-stick surface to any protein. We have neutravudin, we have an antibody, and then we're going to capture monomer and aggregates of alpha-synuclein. And then we're going to come in with another antibody that has a fluorophore. It's not going to bind to monomer because there's no epitope accessible, but it's going to bind to the aggregates. And then we're going to make this fluorophore blink, and then we're going to be able to image the aggregates. And this is what we see. So this is a 100 nanometer scale bar. You can see these small aggregates that we see in cerebral spinal fluid. I mean, for me, they were surprisingly large. They're sort of the same size as coronavirus, so they're 100 nanometers. And what we find is there are also aggregates present in blood, in serum. Again, these are a little bit larger. And then the other exciting thing that we found is there were clear differences between the aggregates in the patients unfortunate enough to have Parkinson's disease compared to the controls. So the patients with Parkinson's disease tended to have more and bigger aggregates. So we're very excited that this might be a way of diagnosing Parkinson's disease early, so you can intervene earlier, ideally before people even have symptoms of the disease. The other thing we found is these aggregates are very good at causing an inflammatory response, not by the adaptive immune response, but by the innate immune response. And so we built an instrument to study this. So what we have here is a sheet of light that is focused on the surface of a live cell. And then we have a fine nanopipette, which we're going to deliver aggregates to the cell surface. So the tip of this pipette is typically about 100 nanometers. So it's like the pipettes you use at school, but with a finer tip. We're going to shoot out aggregates onto the cell surface, and the innate immune response is going to start, and we're going to watch that process occur in real time on a live cell. So this is just to show you, using the pipette, we're very good at delivering molecules. So this is a real-time video just shooting a molecule, wheat germ agglutinin, that has a dye on it onto a glass surface. So all these spots are just under a micron. And so what we're now going to do is have a protein called MyD88, which is labeled with a protein that you get from a jellyfish, green fluorescent protein attached to it, so we can visualize it. And what happens is the aggregate binds to a protein called toll-like receptor 4, and then you form a mydosome that contains 6 MyD88. So initially you have you just see a uniform fluorescence of MyD88, but when we trigger when the aggregate's delivered to the surface, you see these spots form. So maybe we can start the video again. Okay. And so you can see in real time these midosomes form on the cell surface. And this initiates a whole bunch of biochemistry that leads to ultimately to the production of pro-inflammatory cytokines. So we're doing a lot of effort to understand how the aggregates cause triggering of the innate immune response, which aggregates are most potent, and understanding if we can block this process as a potential cure for Alzheimer's and Parkinson's disease. And I'd just like to finish, I think I have very similar conclusions to Shankar, which is probably a good thing. The idea came from basic curiosity-driven multidisciplinary science. It was a big team effort, and we really need to acknowledge all the people who worked in our labs at Selects and Illumina who made this possible. I think an important aspect of our success was that we talked and socialized as a multidisciplinary team, and I think this is where the best ideas come from if you want to do impactful science. I think you might only be at the right place at the right time once. So we didn't know if our idea was going to work or not, but we took the first steps of at least trying it out. The worst thing that could have happened is it might have failed. And as I've shown you, it actually worked well beyond our expectations. And again, like Shankar, my final conclusion is new chemistry, and particularly new ways to measure things which come from physical science, can solve major societal problems and improve people's lives. Thank you.
Thank you very much. And I think we're getting some very good messages, which I suspect are going to come back throughout the lectures this morning about the importance of fundamental basic science and working in teams and don't give up. And perhaps on the same theme, I think I'd like to introduce our next laureate, Dr. Pascal Meyer. He's CEO of Alphonsus and affiliated professor at the University of Strasbourg. He completed his PhD in macromolecular biophysics at the Institut Charles Sadron on the University of Strasbourg in 1991. His thesis explored the dynamics of DNA during pulse field gel electrophoresis and the creation of associated automated optical instrumentation. Importantly, we're in Canada. His first postdoc was at the University of Ottawa from 1991 to 1994, where he worked on the physics of DNA in gel sequencing. Following a second postdoc in France, he initiated his foundational work on DNA sequencing on self-forming DNA colonies at Glaxo Wellcome's Research Center in Geneva. This work continued at Serono and Mantea Predictive Medicine in Geneva until 2003. And as you've already heard, this work became a key component of today's next generation parallel sequencing. Currently, in 2014, he served as co-founder and CEO of Alphonsus, a startup company using original artificial intelligence algorithms to assemble non-pharmaceutical ingredients into safe and patentable formulas with strong biological activities of medical and non-medical interest. 2024 Canada Gairdner International Awardee Dr. Pascal Meyer will be presenting from next generation sequencing to next generation therapeutics, simplicity as a paradigm for scientific disruption. Please join me in welcoming Dr. Meyer. Disappeared? The title disappeared?
Bonjour, merci beaucoup d'être présent. Par égard à mes compatriotes et cousins québécois et acadiens, je vais commencer en français. But for the sake of being understood by the rest of the world, I will finish in English. As the introduction, as you realize, there are basically two places in the world where this technology was incepted. One you have been presented in Cambridge, and I'm going to present the roots in that I called the Lemanic roots. And Lemanic is because the Geneva Lake is also called the Lac Léman, and the area around is also called the Lemanic area. So the first thing is going back to my background. I originate from a small town in the northeast of France, a coal mining town. And I made all my studies in Strasbourg, where I met, thanks to linear algebra, my wife Mireille, who is here today. And during my thesis, we had our 2 daughters. I started working on DNA technologies during that thesis up to a postdoc in Bordeaux again, going through Canada. And I was touching many different aspects, including instrumentation, computer programming, biology, biophysics, etc., which was the perfect profile for not finding a job, neither in academia nor in industry. So by that time, as has also already been presented, it was really very cumbersome to make sequencing, especially using gel electrophoresis or later capillary gel electrophoresis. It was slow, cumbersome, and I didn't want to spend the rest of my life making gels or running these kinds of instruments. There were some new techniques coming up, but all of these techniques had something in common, is that if you wanted to get one piece of sequence, you needed to manipulate one sample. So if you want 1,000 pieces of sequence, you needed to manipulate 1,001 million test tubes. And radioactivity was much used by that time, for those who remember these days. So while I was desperately looking for a job in summer '96, I had some time also to think a little bit. Still, doing better than gel electrophoresis, and I thought about the different things I did during my path, self-organizing structures, microbiology, PCR that I made at a journal club at some point. So it was a lot of physics and Greek with on macromolecular biophysics. Also single molecule observation, DNA molecule observation by video microscopy. Well, it's not a single fluorophore, there are plenty of fluorophores, but gave me the opportunity to be also, like David, confronted to very sensitive detection methods. And at some point, thinking of something, you have an idea, and then you need to find a place to put it in practice. Luckily, I was hired by Glaxo Wellcome in Geneva, and arrived in November '96. And they wanted me to work on the sequencing by hybridization with radio-labeled probes, and it would have meant manipulating tons of radioactivity. And so I said, okay, guys, maybe we first pay attention to my idea. And the first scientist I was talking to was Laurent Farinelli, with whom we made the first experiments. And the picture I'm showing here is the very first picture of DNA colonies, or DNA clusters, I call them colonies, that is observed here with confocal fluorescence microscopy. They were made on a glass plate, but many things had to be done since then. So the first patents were filed April 1st, '97, by Glaxo Wellcome, and later I'll tell you why it was patented so quickly. And what are in these patents is things that we could only present briefly at a conference, which was named Automation in Mapping and Sequencing, because by that time it was all about robotizing the very cumbersome sequencing technologies. And so I presented this. Shankar left me the honor to present the very complicated, at least to explain way to make DNA colonies. It was basically you make PCR, like the PCR that we all had during the COVID, but instead of doing this in a test tube, you attach the DNA primers and the DNA fragments you want to amplify on the surface. And when you do this, in fact, you realize that different DNA fragments amplify locally and end up making what I call DNA colonies, in analogy to the way bacteria multiply on an agar plate. when simultaneously and independently and also in stealth mode, there was also a solid phase amplification method developed in the USA by a company named Mosaic. George Church was also involved a little bit in this. And so I need to explain very quickly what's different between what we were doing and what they were doing. Basically, when you're making PCR in solution or in the solid phase, you want to check whether a certain sequence of DNA is present in your sample. A lawyer would say you want to know if Paul is in this room. So in doing this, you have two pairs of primers that are specific of the sequence you want to amplify. You make the PCR either in a test tube or on the solid surface. And if there's an amplification, you know whether Paul is in the room, whether your COVID is in your nose or whatever. What I wanted to do is something very different. The idea was to fragment the genome, either randomly or by restriction enzymes, and to tail them with two universal pair of primers. So each fragment of different sequence is tailed by the same pairs of primer. This allows then to amplify them on a surface with only one pair of primer on the whole surface, however large it is, and to get the DNA colonies. And so the answer is not now anymore, is Paul in the room, but the question would be, who is in the room? And I get the identity of every person in the room. That's the idea behind. So back to that famous conference, I presented the stepwise base-by-base sequencing. We later realized that the concept was first presented in a patent by Pepe Ross in Roger's Tsien, a future Nobel Prize lab, but they didn't get the funding to work on this. So it was still available, so I presented the principles and especially how we applied this to DNA colonies. By that time, we had only one-color nucleotides and only on 2 of the 4 bases. So the first picture presents, try to use this, when what happens when we incorporate a C on, as Shankar presented, on DNA fragments with a certain primer on them. The first one is supposed to incorporate a C, but not the second one, and we see DNA colonies appearing. Here it's observed again by a fluorescent microscope. When we then incorporate the T, we see 2 things. One is that the colonies that were present in the first image are now more intense, and it can be quantified. You see the numbers here, it's the peak intensity on each colony. And we see also other colonies appearing. So this showed that we were indeed able to incorporate specifically the 2 bases. And the third image is what happened when we incorporated the C, and nothing basically changes in the images, showing that we were indeed possible to do this. So we also wanted to sequence 10 bases or 20 bases, knowing that we were making the constructs with a restriction enzyme, which gave us more information, 6 base more information, which we can argue that it's enough to position it. And in order to make de novo sequencing, we had ways to prepare the templates such that we could, in fact, sequence fragments of a few thousand bases by bits of 25, and then you have longer reads that could have been done. It's a long story, which is still a debate now. So I presented the performance, but by that time, my manager asked not to put the real expected throughput, only the rigorous extrapolation of what I was showing, which ended up to be 100 times faster than existing methods, but already quite more attractive in terms of cost. But we know already, like Shankar, that the throughput would be enormous, but because by just improving the density of colonies, the time of exposure, a number of things, in reality, we were totally conscious that it would be between 100,000 and 10 million bases per second, and costs around $100. So for those who buy the Illumina kits, you can have an idea. So next thing is, second part of the lemanic route is that I convinced at some point Cyrano to create a spin-off in order to further develop the technology, which needed plenty of different specialties to work together. And we founded Mantea Predictive Medicine, initially Geninex. And our CEO, who was the former head of marketing of Serono, wanted to not only to make the instruments, but he said we need to make simultaneously the instruments and the personalized medicine that goes with it. And we were thinking of personal genome card to be used for pharmacogenetics. Now here are the instruments where we were on summer 2003. You recognize the microscope, and if you would look today in an Illumina machine, you would still find the basis of fluorescent microscope without the observation thing, computer like there were at that stage, pumps, valves, etc., all it takes. And here I present some of the technological details, but I go quick. And just to show where we were in the good days, so we were able to sequence up to 10 bases, and you can follow here on the image that the two first are not supposed to be incorporated. The first one is, we see a signal. We had also our own cleavable terminator chemistry, not as elegant and efficient as Shankar's one, but we had something that was working. At least it was just needed to have more regularity on this. So at this stage, I want to thank all these people who work beside me on getting all this done. It is a long list. You can find it on my LinkedIn profile if you want to check. And I want to stress in particularly Laurent Farinelli, who was the guy I first met at Glaxo Wellcome in '96, and who was he was in the lab already. I barely knew where the coffee machine was. So together we made the first experiments and got the first results I showed earlier. So many things are, how did this end it? But our main shareholder, Serono cash-rich company, decided for some reasons to stop all this. They liquidated us and they sold I guess you can admit it's valuable items to Solexa. As is written in a book, it seems that a few weeks later, you got the first sequencing results and the first VX sequencing thanks on using our instruments that I was told. And then you made a very good business that Serono passed, okay. So what does this technology do? It has been explained, a million times faster, cheaper. Also, this technology was used to identify the presence of coronavirus in the first COVID patients, and this is really due to the fact that you can make this anonymous sequencing of every DNA that's in a sample and not to make a hypothesis on what you are looking for. Then, I take this that way. We had several things also, but to make it short here, there's also, this technology is also used in order to find the genetic roots of disease. And this is one example, and what comes out very interestingly is that you don't get one root. There are several genes linked on strongly linked on certain disease, not talking on those that are less strongly linked. And this means that there's something wrong with a traditional pharmacological paradigm. It says you can cure somebody by having one compound going to one target and have one biological effect. Well, here you don't know which one to choose. So I believe that a revolution is needed in this field, and as you know, in France, we are good at revolutions. So the approach I'm thinking of is can be seen as inspired by the strategy of 1,000 cuts. So what does it mean? That we want to perturbate the cellular system with massively diverse mixes of compounds. So you don't have one molecule acting on a single target, but you have plenty of molecules acting simultaneously on many targets or biological features like membrane rigidity, pH, whatever you can think of. this can look like this when you have the molecules acting. In fact, if one molecule is acting to push the system in one direction, another molecule would act to push it in the other direction, and most often the effect is neutral, but in some more rare cases you can have a strong biological effect while not having eventually deleterious effect. It's inspired from a certain number of work, and I had to publish also some work that gives the statistical biophysical roots of what I'm doing. And in order to reduce it to practice in a tractable way, we are working with mixes of edible plants. So it can, I see all the objections raising in the room, but I can assure you that while it takes more than 1 minute, in less than an hour, I can wipe all these objections out in a convincing way. So good thing is that the regulatory agency encourages us to make such experiments by encouraging us to eat very diverse foods. So there's normally no concern about toxicity on making mixes, and the theory shows that this is indeed the case. We're making water extracts for the chemists who are in pharmacy. When you have a heat, you spend 2 to 5 years in making it water-soluble. By using water extracts, we have already water-soluble molecules, and in some sort of massively parallel supramolecule phytopharmacology. When you come with a new technology, it's always difficult to describe and to give it. So it's and the very important thing is also that this is patentable in contrast to classical phytochemical approaches. Because it's synergistic, you get a composition patent mix, which is very important to later protect the discoveries and the clinical validation. There's an issue, however. If you take a library of around 300 to 1,000 different botanicals, and you want to make mixes of 20 of them, you have a gigantic number of possibilities that no organization can handle exhaustively. So we thought of something, of using AI specialized algorithms to do this, and these are themselves inspired from algorithms we were thinking of in Manteia in order to eventually find the genetic roots of the diseases, because by that time the geneticists were saying, no need to sequence whole genomes in days. We don't know what to do with the data. So we had also to think about ways of analyzing the data. So that's what I basically did. It goes a little bit like this. We have the plants. We have the biological assay in vitro. So it's like in classical drug screening. We make the assays and we enter the data into our algorithms. And this is since 2015, so it's not a portal to ChatGPT. And they propose us potentially improved work methods, and we circle this iteratively in order to get this done. We did this during COVID to find an anti-coronavirus. So we started with the classical experiments on Vero EC cells infected by the coronavirus. Here, each bar represents the effect of one extract, of one water extract of complex mixes of edible plants, as I call them, WECMAPs. And most of them have no or very little effect, but we enter this data in the algorithms, and they propose us potentially improved mixes. We assay them again, and then again, and very quickly we're able to come to mixes that prevent most of the mortality of these cells in vitro. It took us a few weeks during COVID, confinement, and Christmas and New Year. So it's very, very efficient. Then we tested it on animal models. This is a model that was first, the debate was, should we use a model where all the animals are dying, or should we only use a model where only the lungs are infected? We had the model which was available where all the mice die when they are not treated. And here we got 30% survival when they are treated by oral route with our mixes. And these in reality are real survivors. We redid the experiment and show that these ones survive longer. And we see it also on the weight curve that the survivors really recovered. And we wanted to then make a clinical evaluation of this. So we produced the product and registered it as a formal food supplement that we could also sell, and we tried it on ourselves. But unfortunately, we didn't get traction enough in 2021 to make a clinical evaluation of this. No comment. Then another aspect that's important for what we will be doing now, here we see the efficacy of our mixes relative for the reference strain with the different variants that have been tested. And in gray, it's a precision medicine, which I don't cite here. It's not chloroquine, it's another one that is seen as much more valuable. And we see that we kept the efficacy in all the strains, whereas the precision medicine lost its efficacy already in some of the strains that were found in 2021, which is something expected with a single. 1 minute. Okay, so I have 3 on my timer. And we have also a lot of data on bacteria, on resistance profile, which are very good. So here is an update of all the results we have already. And then we are thinking of something, the big picture really is to enable personalized medicine. That is, we would have a database of relating the compositions, the effects, and the AI, and when you have a patient diagnosed and its genetic makeup with this data, we can propose a personalized preventive and curative work map. And before making all this picture, we want to respond to the big threat today that the WHO is announcing, with millions of people dying in the coming years because of bacterial resistance. And we would use our products that are already active against these bacteria in order to have therapeutic boosters that the doctors could prescribe on top of the antibiotics they are used to prescribe, and so they would recover their efficacy on the first prescriptions and not as of today after 1, 2, 3, or 4 prescriptions, where in some cases you can have 20% mortality because of this. So that brought me to the end. So I want to thank especially the Gairdner Foundation, the associated committees, the nominators and supporters, the whole teams, again, that are behind all this work, and the scientific community who is using that technology and making, demonstrated in some sense its importance. And I must say also to all those teachers, mentors, one of them is here today, Professor Gary Slater from University of Ottawa, friends and family, all of these people who forged me and allowed me to make this, I hope, become a reality. Thank you very much.
We're arguing about timers here. Well, obviously we have different time dimensions, but absolutely fantastic, all 3 talks. I hope you've heard a lot about DNA sequencing, and you've understood a lot of the background and also just the journeys of the people. Now we're going to take a break, and we'll be back at 10:40 on my time. Okay, everybody, welcome back. Please take your seats. I am now going to leave the stage and hand it over to a new chair for the rest of the morning. I'd like to welcome Dr. Brad Walters, Executive Vice President, Science and Research at UHN, to chair the rest of the day. Brad.
Well, thank you, Janet. Michel Sadelain is our next international laureate that we'll be able to hear from this morning. Michel is the founding director of the newly established Columbia Initiative in Cell Engineering and Cell Therapy at Columbia University in New York. He previously founded and directed the Center for Cell Engineering at Memorial Sloan Kettering Cancer Center. He received his MD from the University of Paris and his PhD from the University of Alberta. Following his postdoctoral research at the Whitehead Institute at MIT, he joined MSK in 1994 and more recently Columbia University in 2024. Michel Sadelain has made several key contributions to the emergence and success of CD19 chimeric antigen receptor, or CAR therapy. His research has contributed to many facets of CAR therapy, including T-cell engineering methodologies via retroviral vectors and gene editing, CAR design, the dual signaling receptor concept, which became known as the second-generation CAR, the identification of CD19 as an effective CAR target, the T-cell manufacturing process itself, GMP processes in collaboration with Dr. Isabelle Riviere at MSK, and the successful clinical translation for B-cell malignancies, AML, now expanding into other diseases and other solid tumors. Sadelain is the recipient of the Cancer Research Institute's Coley Award for Distinguished Research in Tumor Immunology, the NYI PLA Inventor of the Year Award, the Pisano, Gabi, Pasteur, Wiseman, Servier, and Leopold Griffuel Awards, the INSERM International Prize, the Breakthrough Prize for Life Sciences. He's also previously served as president of the American Society for Gene and Cell Therapy. The 2024 Canada Gairdner International Awardee, Dr. Michel Sadelain, will be presenting CD19 CAR T cells, Paradigm for the Living Drug Concept. Please join me in welcoming him to the stage.
Thank you. That's not, I need the clock. Close it. Okay, okay. Ooh. All right. Thank you very much for the kind introduction, and thank you, first of all, to the Gairdner Foundation for recognizing the work of Zélie Guichard's and my own, and in doing so for recognizing the value of T-cell engineering. So what I'd like to do today is to tell you the story of the rise of genetically engineered T-cells becoming drugs. First, drugs for cancer therapy, and perhaps soon drugs for other diseases. A few of us some 30, 40 years ago were, of course, interested in advancing immunotherapy to treat cancer, but there was, at the time, quite some disbelief that the immune system could ever succeed in overcoming cancer, and that is because so many of us develop cancer or will develop cancer and succumb to that disease despite having our immune system. Before I get into what led us to want to engineer T cells, I should give you a little bit of background on our immune system. Obviously, our immune system is extraordinarily valuable in keeping us alive and fighting the entry of foreign parasites into our body, may they be viruses or bacteria. And evolution has given us very incredible tools to fight the entry of these parasites, of these undesirable parasites. In what's called the adaptive immune response, we have both T cells that have exquisite specificity to engage molecules that they recognize as not belonging in our body, as well as antibodies, which are produced by B cells. And to build up this line of defense, in the case of T cells, our thymus produces these cells that in blind fashion assemble a set of genes called V genes, D genes, and J genes that produce a collection of T cells that we refer to as a repertoire that is ready in our body to fend off one of those invaders should it come to the body at some time in our lifetime. Some scientists think that we have about 20 million or so of these different T cell receptors in a normal adult. However, in the case of cancer, the invader is not foreign. It comes from us. It is one of our own cells that begins acquiring somatic mutations, and in doing so acquires new properties of invasion, proliferation, failing to die, and immortality. These mutations can sometimes be recognized by the immune system, and in fact the immune system shapes the development of cancer, something that is known as cancer immunoediting. And in some patients, we can use antibodies that will unleash the power of T cells that are formed in some patients that can eliminate these cancers, but in many more, this natural immune response, if you like, is insufficient to prevail over cancer and eliminate the cancer. And for this reason, this led some of us, a handful of us, some 30 years ago or so, to think that we needed a different approach, or at least another approach. And this approach would rely on providing an instruction to the T cells, an instruction to guide the T cell to the cancer, and to go on to kill those cancer cells. And so the field of CARs was born sort of on the fringes of other fields, on the fringes of, on the upper right here, of tumor immunology that has taught us so much about the biology of T cells, the microenvironment, the targets that exist in cancer cells. But it's also different from conventional immunology insofar that these T cells will be repurposed and reprogrammed, and to do this, we need genetic tools. The first genetic tools that were used were retroviral vectors, and today we use more sophisticated tools such as CRISPR to edit the genome of T cells. New receptors had to be created, synthetic receptors that would engage different targets in different ways, and I'll come to that, of course. Those are the molecules that some years ago we named CARs. And these receptors, unlike the physiological T cell receptor that engages what are called HLA peptide complexes, differ in their nature. They have to be on the surface of cells. They can be proteins, glycoproteins, but they could also be a carbohydrate or a glycolipid. And I'll come back to the CD19 target, which is really the poster child, if you like, for this therapy. Because so few were in the scientific world and medical world believed that this approach could ever succeed or be useful, the few of us working on this had to prove it beyond animal studies and bring it to the clinic. And that obliged us to really become cell manufacturers, something that started not in the industry, but in academic centers. And of course, in doing so, the process for making these cells couldn't be that you would use in the research lab, but would have to follow a set of rules set forth in the United States by the FDA. So how to summarize the first 15 or 20 years of this in one slide, I will attempt to do so. When I left the University of Alberta, I wanted to engineer T cells, and there weren't so many places in the world where the rudiments of gene transfer to mammalian cells were being established, and one of those was MIT at the Whitehead Institute. And so I went there to learn the basic principles of genetic engineering at the time. And it took me a few years to come up with this demonstration here that we could take real T cells, primary T cells, in this case from a mouse, and genetically modify them using recombinant retroviruses carrying, in this case, the E. coli LAC-Z gene. This is before even the GFP protein that was mentioned earlier this morning had been discovered. This didn't elicit any interest back then when I presented this for the first time at a World Congress in Budapest. And yet, for me and those working with me, this was a magic moment where we thought that we had now a path to go and engineer T cells. And an important decision that we made in retrospect, a very critical decision, was that we would study T cell signaling not in what are called leukemic cell lines or hybridomas, as so many others did and taught us many things, but that we would study T cell engineering and T cell signaling in authentic primary cells. And I think it's very largely thanks to that approach, which is admittedly cumbersome and slower than other methods, that we came to uncover molecules that were actually functional in human T cells and ultimately in patients. We had to design new receptors, and here I refer to the cloning of the zeta chain, which is one of multiple chains that T cells use to initiate their activation. 3 groups in 1991 independently cloned this chain and created these fusions receptors with extracellular domains that you see here, because this zeta chain is really an intracellular protein. And in doing so, they found that this zeta chain was sufficient to initiate the activation of a T cell. And then Zelig Eshar, whom I want to name because he's my co-recipient today, introduced this variant on this zeta chain fusion by fusing here elements derived from an immunoglobulin, the heavy and light chains of an antibody, which confer the specificity, the exquisite specificity to different antibodies. And since he's not here, here's a picture of Zelig, whom I last saw in Tel Aviv in February of 2020. But because we could study genes in primary cells and not just in leukemic cells, we realized that these zeta chain fusions were not that effective in redirecting an immune response. You could get a T cell to kill maybe a target once, and then the T cell would collapse in a process known as activation-induced cell death. And so we studied that signaling pathway for quite a while and eventually realized that if we started combining signaling domains within these molecules, we could now achieve or create, if I may say, T cells that could engage an antigen, kill the targets, the cells that express that antigen, but also go on to proliferate and kill more cells again and proliferate some more and kill again. And this was obviously what we were looking for and the necessary, the obligatory path to come to what we later called a living drug. We needed to identify a target, first for our experimental work, and one that we thought we could eventually bring to the clinic. And we focused on this molecule called CD19. CD19 was known as a molecule found in B cells, cells of the immune system, from which different leukemias and lymphomas occur, B-cell lymphomas and acute lymphoblastic leukemias. And CD19 was not a very popular molecule at the time. It was known to participate in B-cell activation. But we thought that it would be a good target because it is expressed in the majority of these cancers. It is in the body found only in B cells, and even if you destroyed those B cells, well, patients could still fare well. And in 2003, we provided this first proof of concept experiment showing that now you could take human peripheral blood, just from a blood draw, engineer them with a recombinant retrovirus that encoded a synthetic gene that itself produced this synthetic protein that we called a CAR, and the CAR recognized CD19. And if we infused only once these T cells into mice that bore either leukemias or lymphomas, we could eradicate the cancers in those mice. And this led to gave us the impetus to proceed to clinical trials at my center. And thanks to my colleague Isabelle Riviere, who joined us from MIT, we were able to build this facility and establish processes that would allow us to manufacture peripheral blood T cells drawn from patients for infusion to those patients. I won't walk you through the early clinical studies. There were 3 teams that produced clinical results at about the same time. Ours, the National Cancer Institute, to whom we had provided our CAR molecules, and also the University of Pennsylvania that had received their CAR molecule from another group. So I'll just illustrate what these CAR T cells do from one of our earlier papers, the first one in acute lymphoblastic leukemia. And so these are patients that have a relapsed refractory disease. They've already received a number of chemotherapies, some of them a bone marrow transplant, and unfortunately, when their disease relapses and is refractory to any treatment, they only have a few months to live. And so here are subjects 4 and 5 from the first 5 that we reported on. And what you see here are leukemia cells that express CD19 in bone marrow samples a few days before receiving the CAR T cells. And you can see here 10 days or 11 days later or 2 weeks later, the complete absence, the complete eradication of any leukemic cell in the bone marrow of these patients. You couldn't detect them by sensitive methods. So these are called molecular responses, molecular complete responses. Later that year, the journal Science proclaimed cancer immunotherapy to be the scientific breakthrough of the year. That was largely for checkpoint blockade, another very successful immunotherapy, sort of an older big brother to CAR therapy. But the editors already saluted, if I can say, the emergence of this approach in 2013. The trials conducted at our center and others were soon confirmed in larger series And by 2018, soon after the approval of these drugs by the FDA, Carl June and I summarized all this literature that clearly indicated that these results could be reproduced in the hands of many. Here are the first 2 CAR molecules that were approved by the FDA in 2017. Both of them are specific for CD19, that the heavy and light chains of the immunoglobulin here in the extracellular domain would bind to. Both of them have this dual signaling motif that I mentioned before. One of them uses CD28. This is the signaling domain borrowed from an important costimulatory receptor in T cells known as CD28. The other one uses the signaling domain of another costimulatory receptor known as 4-1BB, a member of the TNF receptor family. There's a lot to say in comparing these two, but suffice it to say that they achieve fairly comparable results in comparable clinical outcomes, I should say, in patients with leukemia. And so there are today 6 CAR therapies that have been approved by the US FDA. 4 of them target CD19, and 2 of them target BCMA, another target of interest in the disease multiple myeloma. And all of these CAR therapies were first tested in patients with relapsed and refractory disease and are gradually moving into earlier phase of treatment of these patients. But in those that have relapsed refractory disease, this is the complete remission rate that you obtain in different diseases. In some forms of B-cell lymphomas, such as mantle cell lymphoma, follicular lymphoma, as you see, 80% of these patients will go into a complete remission, a bit less in the case of large B-cell lymphoma, and in acute lymphoblastic leukemia, a remarkable 80% or more of these patients will go into complete remission, and about half of them will survive in the long term without requiring any further treatment, but the other half will still at some point relapse from their disease, telling us that there's still progress to be made. And so I'd like to spend the next few minutes by telling you, giving you an insight into some of the directions that we're pursuing. And so we have this foundation now, these CD19 CAR T cells that are useful in a series of B-cell malignancies, but there's room for improvement, and of course we want to expand this approach to other cancers and other diseases. I would like in the next few minutes to touch upon some directions for future work. Can we create a more sensitive T cell? And I'll explain what that means in a minute. A more discerning T cell that could tell the difference between a cancer cell and a normal cell. Can we design T cells that are both potent and durable? Often the more potent cells don't last so long, whereas those that are endowed with long-term persistence are not necessarily as potent. And lastly, mentioned briefly, can we actually bypass the need for a human donor for these T cells? And you'll see what I mean in a few minutes. So first, a few slides just to illustrate this issue of antigen sensitivity. Clinical reports taught us that sometimes patients relapse after a CAR T cell therapy with the target antigen still found in the tumor. So why is it that the CAR T cell is failing to eliminate those cells? There are a number of reasons for CAR T cells not succeeding in eliminating a tumor. They may not migrate and enter into the tumor site. They may be suppressed at the tumor site. But in some cases, we know that the T cells got there and the tumor is relapsing, but with a lower level of antigen. And that led us to examine the sensitivity of these CAR T cells. And here I should bring in the physiological receptor, the T cell receptor that I mentioned earlier. It is depicted on the left here. And this is a complex that aggregates many chains, the alpha and beta chain that recognize the HLA peptide complexes, and then a variety of chains here that together initiate activation of T cells. And this receptor is known to be highly sensitive, meaning that at least under ideal conditions in vitro, a few molecules of the HLA peptide complexes are sufficient to trigger activation. But when we started looking at these CAR T cells, we found that they need about 1,000 molecules, or of course more, 1,000 molecules to initiate successful activation and then killing of their target. So that led us to create this hybrid structure that you see here, where we preserve the antibody-derived elements, the heavy and light chains, and insert them into this T-cell receptor complex by swapping out the variable regions with these antibody-derived elements. And by establishing panels of tumor cells where we control very closely the number of target molecules at the surface of those cells, we could study the sensitivity of these new receptors. So you see a natural acute lymphoblastic leukemia may have 20,000 or more CD19 molecules on its surface, but we have lines that have 2,200 or even few. The short of it is that the HIT receptor is a lot more depicted here, which is an HLA-independent T-cell receptor because it engages antigen without the need for HLA, is far more sensitive than the conventional CARs. And I can show you a few examples of that. Now, if we create an acute lymphoblastic leukemia that we know has 200 molecules of CD19 on its cell surface, the best conventional CARs we could use fail, whereas the HIT receptor will succeed in these mouse models. Likewise, in models of acute myeloid leukemia, the best CAR that we could come up with targeting a molecule called CD19 fails in the presence of that low number of target antigens, but the receptors succeed. And another example, not yet published, targets CD70 in a model of kidney cancer. And here too, in the kidney cancer, the best CARs that we can design will at best induce a stable disease. You see that when you look at the tumors in these animals, very much like what you see in patients. Some of the cells stain positive for the antigen, but many others appear to be negative. In this instance, they're not negative, actually. It's just that they express very low levels of the antigen, and the HIT receptor is capable of eliminating these cells. The next topic I want to address quickly is, can we make sort of try to make a smarter T cell that is more discerning or selective in terms of its of the targets that it engages? And here I have to introduce some terminology that many of you will remember from your algebra, Boolean logic terminology. So unlike natural T cells that have one receptor that engages one HLA peptide complexes, we can, of course, design T cells that could engage two antigens or more. But if you think about how to engage two molecules, there are many ways to do so. You could have a T cell that recognizes antigen A or B, call that an OR gate. You could have what's called an AND gate, where a T cell would be activated only in the presence of A and B being together on the surface of T cell, but not being activated if a cell expresses either A or B alone. There are also NOT gates. We created some inhibitory receptors to antigen B. In this case, the ideal goal would be a T cell that would kill tumor cells that express A but not B, but to protect a normal cell that expresses the antigen B, triggering the inhibitory receptor. Another gate is called an if-then gate, which introduces a temporal dimension where a receptor, not a CAR, engages antigen A. This is typically the Notch receptor, which then initiates the activation, the transcription, of a CAR that engages antigen B. So in this instance, the CAR recognizing B is only expressed wherever in the body where there is antigen A. And lastly, the one I want to discuss is an if-better gate, which is, can we have a molecule B that would help recognition and killing of cells that express A when A is of limiting abundance? And let me walk you through what we're doing in the case of acute myeloid leukemia. And this is a survey of 35 patients at Sloan Kettering looking at the level of expression of different molecules on these leukemias. One's called CLEC12A, another one AGRA2. Here's another one called CD123, and the label's missing here, but it's CD33. The Y and X axis here plot the median number of those molecules found on the surface in those patient samples. You might think that CD123 and CD33 are quite attractive because they are more abundant, and indeed there are today several trials targeting these antigens. But we were a bit fearful of doing so for the reason that's shown here. In normal bone marrow cells, these antigens are also expressed. And CD123 and 33 are found on many cells, including hematopoietic stem cells and others, which means that if your CAR T cell could not distinguish the leukemia from these normal cells, you wouldn't just eliminate the leukemia, but you would destroy the recipient's hematopoietic system. So we wanted to explore whether we could try to be more selective towards these cells while sparing the cells that express low levels of AGR2, which we know are found on hematopoietic stem cells and what's called common myeloid progenitors. So we designed this dual system where a CAR recognizes antigen A, which is AGR2, and this helper molecule targeting antigen B, which is CLEC12A in this case. And I'll just show you what we can do now in vivo in animal models. So on this spectrum of patient samples here, here's a cell line that expresses, you see, 10,000 molecules of AGR2 and 500 of the helper B molecule. So we're positioned here. We put these leukemias in mice and treat them with CAR T cells that are engineered with these two receptors. And it's a low dose of T cells, but this CAR is remarkably effective and the leukemia is eliminated. You might ask, well, did the antigen B matter in this case, which was this one? No, it doesn't. In fact, if you eliminate it through genetic ablation, which means that now you have dropped your position here on the spectrum to this one here, these CAR T cells still work. And they work because there's 10,000 molecules of AGR2 at their surface. But now, let's go lower. And now we have a leukemia variant that has 1,000 molecules of AGR2 on its surface. So we are now positioned here. And now we treat these mice and the CAR T cells fail, which is what we wanted in this case, because we want to spare those hematopoietic stem cells. So now is the real test of the if better gate. So now the same cell that has 1,000 molecules of AGR2 has a high level of the second antigen B, and now these same CAR T cells work again. So this is the if better concept. The T cell engages this antigen at this level depending on the context. In this context, it does not, and in this one, it does. Lastly, I want to, thirdly, my third example is how we can improve the functional persistence of CAR T cells. And here we have to go back to the very initial design of these receptors. The first one we reported in the early 2000s that combined this activation domain from this zeta chain from the CD3 complex and the signaling domain of this costimulatory receptor called CD28. The clinic taught us that these cells are powerful, but relatively short-lived. And after a while, we wondered if, in fact, we had created a signal that is, in fact, too potent, too strong. And we thought we should attenuate it and then see what happens. And so we started mutating these domains that are well known in the zeta chain that mediate T cell activation and created a family of these derivatives. And the short of it is that one of them, called 1XX, which has a reduced strength of activation, produced far better results in, again, these leukemia models. And so we brought this to a clinical trial with my colleague, again, Isabelle Rivière, and our clinician, Jay Park. We designed this trial with the following reasoning. If these cells are really more functional or for a longer period of time, meaning if they are superior T cells to what we were making before, then we ought to be able to treat patients with lower T cell doses. And I want to mention here the doses of CAR T cells that are used with the approved clinical products today. One of them, using the 41BB CAR T cells, requires the infusion of 300 million CAR T cells. Another one that uses the CD28 CAR design, which is a bit more potent, requires about 150 million of these CAR T cells. But we thought that if these cells are really better, we should be able to treat patients with the same disease, in this case refractory diffuse large B-cell lymphomas, with lower doses. And so we started treating patients with only 25 million CAR T cells and then dose escalated. And I don't have time to go through the details, of course, so just the answer is simply that we got excellent results already at 25 million CAR T cells. More T cells didn't really make it any better. So we expanded the cohort at this low dose and found that we could get responses that match any other CAR T cell that already exists, but with a 10 times lower dose. And so this is just to exemplify. We're not going to reduce the cost of sequencing by 1,000,000 times, but at least we're reducing the T cell dose already tenfold, and I think it's going to go further than that. We are, today, things are very different from what they were just 10 years ago when a limited number of laboratories were developing these receptors. There's now really an explosion of the variations, if you like, on these card designs, and it's really wonderful to see. Something else that is happening is that work has inspired many to explore not just the alpha beta T cells, the conventional peripheral blood T cells that we utilized up till now, but to start engineering other cells of the immune system, macrophage, natural killer cells, even granulocytes and eosinophils. And here I want to make a comment on the sources of T cells. Everything I described to here in patients makes use of the patient's own T cells. This is autologous therapy. Some have said that it might disappear, it might be replaced by cells that you could make ready-made for infusion to patients, but that's not so easy to do. Allogeneic cells, I'll come back to that in a minute. I think autologous manufacturing is here to stay. The processes used to take 2 weeks, then 7 days, and today many protocols take 3 days, and some of them just emerging are only overnight processes. But there's a lot of interest nonetheless in using allogeneic cells from either healthy donors or cells that never were formed in the body of an individual. And that is what we might hope to achieve by using pluripotent stem cells. And you can imagine that if you have these cells growing in bioreactors, and if we mastered the differentiation process to create functional T cells, that this could actually ultimately bypass the need to go and harvest T cells from humans, including either healthy donors or patients themselves. And I have to stop very promptly. So I'm not going to highlight that there is progress in this space, but there's still a lot that we need to learn to generate authentic T cells in a dish outside of the thymus. Some of the last things I want to comment on is the expanding applications of this approach. So CD19 CAR therapy, as you can tell here, is the tip of the iceberg. There are today BCMA-targeted therapies for multiple myeloma. AML is another important leukemia for which a solution is still not found. And of course, we all await success in the variety of solid tumors. But there's already wonderful applications that are coming up in autoimmunity using these very same CD19 CAR T cells, treating patients with systemic lupus, with other encouraging results in pemphigus and other settings. In animal models, CAR T cells are starting to be used to remove, for example, fibrosis in the heart after infarction, or removing the accumulation of excessive senescent cells. My almost last comment is that really CAR T cells are disruptive in many ways. They're not conventional drugs. They're not chemicals. They're not proteins. They're actually cells. Furthermore, they are genetically engineered cells. And furthermore, they make use of synthetic genes to reprogram those cells. But the novelty and challenges that it raises extend beyond that. For the pharmaceutical industry, obviously they have to put into place a whole new manufacturing and distribution system that didn't exist until now. This is also a challenge for, reimbursements of these cell therapies that are more expensive than standard drugs. We all wrestle now with how to address the issue of equitable access, but this is going to impact the medical practice and therefore what is taught in medical schools in many ways. I just want to highlight one very crucial aspect of this, which is making these cells, producing these cells. The traditional model is that we leave that to the pharmaceutical industry. But that may not be the case for cell therapies. This field was born in academia. You might say it could just transition to the pharmaceutical industry. But I believe that there are so many diseases, some rare and some not, and so many variations on these cell types, the signaling modules, the way you produce the cells, that academia will continue to play a fundamental role in driving this field. And that gives a new mission to medical schools, one that we traditionally give and pass on to the pharmaceutical industry, and that is manufacturing. And our center demonstrates, and many others too now, that you can do that. You can build the facilities, establish teams, develop processes that empower physicians and researchers at their medical center to develop these medicine. So my final note really is that there's so much to do in the world of T-cell engineering, as you can see here. There are so many to thank as well. My mentors, at the University of Paris, at the University of Alberta, at MIT, all those that I worked with, including Anton, who's now a postdoc here in Toronto, who worked with me for the past years. There's one person I want to highlight, and that's Isabelle Rivière. You already heard her name. Because she established at our center the processes that enabled the clinical translation of our research. And without that, we would still just be publishing papers, and this would not have seen the light of day. Thank you.
Thank you. It's now my pleasure to introduce Dr. Laura Evgen, who was selected as a 2024 Canada Gairdner Early Career Investigator by Dr. Sadelain. Dr. Laura Evgen is a scientist at the Genome Sciences Department at the BC Cancer Research Institute and an assistant professor in the Department of Medical Genetics at the University of British Columbia in Vancouver. She earned her PhD in biochemistry from the University of Ottawa under the guidance of Dr. John Bell, followed by a postdoctoral fellowship at the Mayo Clinic in Rochester, where she worked with Dr. Richard Weil. In 2020, Dr. Evgen established her lab at the BCCRI, focusing on enhancing the efficacy and accessibility of T-cell therapies for cancer. Her lab leverages diverse tools and model systems to design innovative CAR constructs, develop CAR T-cell engineering strategies, and evaluate CAR T-cell function in immune-competent models of cancer and autoimmune diseases. Dr. Evgen will be presenting Engineering Effective and Accessible CAR T-Cells. Please join me in welcoming her to the stage.
Last speaker was a little bit taller. Okay, so good morning. Thank you very much. This is a real honor to be here today. I'd just like to start by congratulating Dr. Sadelain and Eshar, as well as all of the other laureates on their award, and thanking them for their really important contributions to science. Okay, so T cells patrol the body searching for signs of infection or cancer. They do so using their T cell receptor that interacts with the MHC molecule that is expressed on virtually every cell in the body and express small bits of peptide of the proteome. When a T cell encounters its cognate ligand, it produces cytokines and other cytotoxic molecules that enable cell lysis. And together, it's really this specificity, functionality, and memory properties of T cells that allow them, or that endow them with very strong agency to recognize and eliminate cancers over time. Importantly, we can also help T cells to focus their attention on cancer cells by engineering them with this synthetic receptor called a chimeric antigen receptor, or a CAR. So as Dr. Sadelain just outlined, the CAR brings together the recognition capabilities of an antibody with the signaling properties of a T cell receptor. The first iterations were developed by Dr. Zelig Eshar and fused the variable heavy and light chains of an antibody with the constant TCR alpha and beta chains of the T cell receptor. This was then simplified to directly fuse the scFv to CD3 zeta. And in both these configurations, a T cell expressing these new receptors was conferred a new specificity for surface-expressed antigens in an MHC-independent manner. However, because effective T cell responses require not only signal 1 in the form of TCR stimulation, but also signal 2 in the form of co-stimulation, it was really the addition of this co-stimulatory domain by Dr. Sadelain and his team that overcame the apoptosis and dysfunction that was associated in primary T cells and really turned this into a therapeutically useful molecule. They also selected CD19 as a target antigen, and this was important because it enabled the clinical translation in the absence or with a manageable toxicity, on-target, off-tumor toxicity profile. So together, the development of the CAR was really opened up this field of synthetic biology and synthetic immune receptors and provided the motivation to develop manufacturing and regulatory frameworks for engineered cell therapies. So CAR T cells have been enormously successful for hematologic malignancies. There are now 6 FDA-approved products targeting CD19 or BCMA for the treatment of acute lymphoblastic leukemia, large B-cell lymphoma, and multiple myeloma. CARs targeting CD19 are now an established therapeutic modality. They can induce prolonged remissions in patients with B-cell malignancies with minimal long-term toxicities. Although they were initially approved in the third-line setting, it's now been shown that they can be an effective alternative to autologous stem cell transplant in the second-line setting for patients with early relapsed or refractory large B-cell lymphoma. There's also now more than a decade of follow-up that has elapsed in some of the early patients that have been treated. And it's really remarkable to see that some of the early patients who are treated still have circulating adoptively transferred cells in their body that continue to protect against relapse. So the overarching goal of my lab is to increase the accessibility and effectiveness of CAR T-cell therapy for both oncology and autoimmunity. There are many levels of scale over which to design and optimize these synthetic receptors that are engineered into T cells and which need to operate in both the micro and macro environment of an individual with cancer or autoimmune disease. And to that end, we've invested in the development of immune-competent mouse models in order to reverse translate some of the clinical observations, as well as assess complex cellular dynamics in the setting of combination therapies and evaluate novel ways to engineer these CAR T cells directly in the body. So there have been many clinical correlates of response in the setting of large B-cell lymphoma, and I'd just like to highlight one, and that is the presence of extranodal disease. So as you can imagine, lymphoma primarily exists in lymph nodes, but in about 1/3 of patients, the disease occurs elsewhere in the body. This can either be as a primary event or as a manifestation of advanced disease. And so there's now several studies that have retrospective studies that have documented that patients treated with axi-cell or teza-cell and who have substantial extranodal disease burden fare worse than those who have fewer or no sites of extranodal disease. And we're looking to recapitulate this in our mouse models of lymphoma. So when we treat our mice that have lymphoma with either 4-1BB or CD28 co-stimulated CD19 CAR T cells, we can dramatically reduce the levels of malignant B cells in these mice. But at this dose, we see that the mice ultimately succumb to disease. At necropsy, we see that the disease is relatively well controlled in the lymph nodes and the spleen, but we see substantial disease burden in the liver and in the ascites. So over time, the efficacy of B-cell clearance varies across anatomic site, and this is shown in the CAR T-cell to B-cell ratio. Particularly, there's this difference between lymphoid and non-lymphoid tissues. And this has led us to hypothesize that there are different barriers that exist within these anatomic sites which underlie differences in functionality of the CAR T cells. And so to explore this, we've taken on 2 different approaches. The first is to design several spectral flow cytometry panels to characterize the tumor microenvironment as well as phenotype the CAR T cells over time and across anatomic sites. And we've seen this very rapid tissue imprinting on T cells. We hope to use this information in order to better arm CAR T cells to perform better at these sites of refractory disease. We've also taken on an unbiased approach where we conducted an in vivo CRISPR knockout screen using a small panel of 120 transcription factor guides to identify genes to either knock out or overexpress in CAR T cells to enhance their function. We've identified a number of candidates that we think would globally ameliorate CAR T-cell function, and some other ones that we think may be more site-specific. And we're in the process of validating these different candidates. Stemming from my work as a postdoc, we're also very interested in how we can leverage the native T-cell receptor in CAR-modified T-cells in order to enhance the function, engraftment, and persistence of these cells, particularly in solid tumor models, now using an mRNA vaccine. And so in our system, we have OT1 transgenic T-cells spiked into a wild-type pool, transduced with an EGFR-V3 CAR, and these cells are then boosted in vivo with an OVA encoding mRNA vaccine. And I'll just point out that our tumor model does not have OVA in this, so this is an irrelevant antigen. Provision of the vaccine is able to substantially increase the global level of circulating CAR T cells in the blood as well as in the tumor, and these OT1 T cells really overtake the population. We also elicit a shift in the functional profile, specifically in the OT1 CAR T cells. There we go. I just wanted to point out that although the CAR was designed in order to mimic the TCR, we think that this is a really interesting way to engage alternative signaling, alternative differentiation programs and signaling in order to maximize their effector function, particularly in the solid tumor setting. So CAR T-cell therapy has really shifted how we think about treating CAR T-cells at the global level, how we think about treating disease at the, with engineered cells globally. There are more than 1,600 trials listed on clinicaltrials.gov. The vast majority of these are for cancer indications. However, in recent years, there's been intense interest in using these cells for autoimmune diseases, where CD19 and BCMA-targeted CAR T cells have been repurposed to treat diseases where B cells and plasma cells elicit autoimmune pathologies. And so we're also very excited about this, and we're collaborating with a number of different groups at UBC in order to apply our CAR T cell expertise to mouse models of systemic lupus erythematosus, MS, and type 1 diabetes. So all of the commercially approved CAR T cell products and a majority of cell therapies that are undergoing clinical testing are made from a patient's own T cells ex vivo. Very, very briefly, the T cells are collected via leukapheresis. They're stimulated in vitro, transduced with a viral vector encoding the CAR, and then expanded to generate a engineered cell product comprising hundreds of millions of modified T cells. As you can imagine, this is a complex process, and the list price for the commercially approved CAR T cell products surpasses $500,000 US. This creates an equity and accessibility barrier and has significantly slowed implementation, particularly in the Canadian healthcare system. So this has provided the motivation to develop strategies to directly generate these cells in the body by direct infusion of either lipid nanoparticle encoding lipid nanoparticle formulated nucleic acids or targeted viral vectors. And we have been collaborating with a number of groups at UBC in order to optimize the design elements for an LNP RNA delivery system for this CAR instruction. At the foundation of this, we think that we need a delivery system to efficiently transfect T cells or other effector cells. And to this end, we've been collaborating with Dr. Peter Cullis and Nanovation to use their long circulating LNP formulations, which have enhanced transfection of immune organs and reduced reduced hepatic tropism. We're also interested in how we can optimize the duration and intensity of expression through mRNA cassette design optimization and choosing ideal RNA species. Finally, at the top of this mountain summit, we're also interested in how we can optimize the payload through CAR design in order to choose designs that will maximize CAR T cell functionality over this very transient expression period. So to conclude, I'd just like to thank the members of my team at BC Cancer and UBC, our many collaborators, because this really is a team effort, and as well my past and current mentors. So thank you very much.
Thank you. So this year's Momentum Award winner is Dr. Megan Azad. Dr. Azad is a professor of pediatrics and child health at the University of Manitoba, a Tier 2 Canada Research Chair in Early Nutrition and Developmental Origins of Health and Disease. She holds a PhD in biochemistry and medical genetics and an MSc in epidemiology. Dr. Azad's research is focused on the role of infant nutrition and the microbiome in child growth, development, and resilience. She co-directs the Manitoba Interdisciplinary Lactation Center and leads the International Milk Composition Consortium. Research in her Thrive Discovery Lab is funded by the Canadian Institutes of Health Research, the US NIH, the Canada Foundation for Innovation, and the Bill and Melinda Gates Foundation. Dr. Azad is a fellow of the CFAR Humans and Microbiome Program and an elected member of the Royal Society of Canada College of New Scholars. She has received the International Human Milk Genomics Mid-Career Investigator Award and was named among the top 100 most powerful women and Canada's top 40 under 40, and received the prestigious Stacey Prize for research in the natural sciences. Dr. Megan Azad, the 2024 Canada Gairdner Momentum Awardee, will be presenting her presentation on human milk, the original superfood, and personalized medicine.
All right. Thank you for the very kind introduction. It is my pleasure to be here and share with you some of our research on human milk. And it's, I think, a big win for this field that we're on the same stage as next generation sequencing and cancer immunotherapies. So that for me is a hugely exciting honor. So in the next 25 minutes, I'm going to share with you why I think human milk is the ultimate personalized medicine. This is a buzzword we hear a lot about. A quote that I love that has inspired me greatly is this one. Human breast milk is not only a perfectly adapted nutritional supply for the infant, but probably the most specific personalized medicine that he or she is likely to receive, given at a time when gene expression, and the microbiome, I added that part, is being fine-tuned for life. So this quote comes from Cesar Victoria, who led the Lancet series on breastfeeding in 2016. This was a time when I was just getting started, finding my ground, and deciding what I wanted to focus on. And actually, he was given a Gairdner Prize in 2017, the year after. And that really inspired me to continue in this field. I actually paid my own way out here to Toronto for Gairdner Week in 2017, specifically to hear Dr. Victoria's lecture and to meet him. And so you can imagine what a surreal feeling it is to be here just 7 years later on the same stage. I think Dr. Victoria and I would have a similar motivation for studying breast milk. And it's shown very beautifully in this map. So you can see here that in some countries in the world, up to 18% of infant deaths are attributable to suboptimal breastfeeding. That's huge. That means that by supporting breastfeeding, we could save many lives. It's been estimated in that Lancet series that over 800,000 lives a year could be saved if we were able to support optimal breastfeeding around the world. Of course, we live in Canada, and that's nice and dark blue, meaning that not many babies here are dying because of suboptimal breastfeeding. So that's a good thing, but it doesn't mean it's not still important to understand and study breastfeeding in the context of Canada right here at home. This is a pamphlet that we give out to pregnant moms in my home province of Manitoba, outlining 8 great reasons to breastfeed your baby. And I won't go through them all today, but as a health researcher, I'm most interested in number 3. Breastfed babies are healthier. For example, they have a lower risk of sudden infant death syndrome, of respiratory tract infections, of type 1 diabetes, and if we look later in life, of obesity into adolescence and adulthood. So as a basic scientist, I want to know why. Why is it that breastfeeding confers all these health benefits? And I also want to point out that the answer to that question can help all babies, no matter how they're fed. And by helping all babies, we can help all families and all societies be healthier. So to answer that question, we've got to think about the milk. I mentioned it's a superfood. Another overused buzzword, perhaps, but I think if one food deserves that label, it is human milk. contains all the nutrients a baby needs to grow and develop for the first months of life. But it's more than a food. We could think of breast milk also as a baby's first vaccine. So a new baby has a very immature immune system. It can't generate the antibodies it needs to fend off infections. It gets that from its mother's breast milk. We talked about personalized medicine. Here's a picture for you. This is breast milk from the same mother a few days apart. You can tell with your eyes that it's a different color. And if you looked at it with a microscope or other technologies, you'd see that the molecules in there are very different too. That's because the milk is adapting to the needs of that baby when it gets sick, which is incredible. Breast milk is also dynamic, so it doesn't just change if a baby gets sick. It changes day by day, month by month as the baby grows and its needs are changing. It even changes from the beginning to the end of one feeding and from morning to night. So you can imagine how that would be difficult to replicate in a one-size-fits-all artificial formula. Human milk is also tailor-made for tiny humans. We don't think about that, but all mammals make milk. That's actually what defines us as mammals. But the milk for every species is different. It's optimized for the babies of that species. So if you think of a baby human and a baby cow, they are pretty different. A baby cow is up wobbling around in the first hours of life, so it needs milk with high protein to build muscles. Whereas a baby human is not walking around for months. It's actually developing most of its brain and devoting its energy to that purpose. So human milk is high in specific fats that are important for brain development. We could talk all day about milk for different species, but I will move on. And comment also that it's not just optimized for the baby, but also for the baby's microbes. So I studied the microbiome using actually next generation sequencing technology. We sequence microbes to understand who's there. And so we think of ourselves as full of organs, a heart, lungs, intestines. We actually have millions and trillions of microbes on our bodies. We actually have more microbial cells than human cells on our bodies, and they're doing lots of really important things. We need our visible organs and our invisible microbiome working together for us to function as a complete human. And we think a lot about the gut microbiome. This is where the majority of our microbes live, and they do very important things. They help train our immune system to understand what we should react to and what we should not. They make vitamins that we can't make on our own. They fend off the bad bacteria, and many more things. So it's maybe not surprising then that the microbiome and disruption of the microbiome is linked to gut diseases like colitis, but not only gut diseases. The more we learn about the microbiome, the more we realize it's actually linked to almost every health outcome, including cancer and liver disease and lung disease. So if human milk is feeding that microbiome, I think that's pretty important. Maybe it's even a 9th great reason to breastfeed your baby. And so this is some of the research that I did as a postdoc, my very first paper on the microbiome, where we showed that babies who were breastfed had a different microbiome. And a few years later, this paper came out from a larger study from colleagues in the Teddy study, and they looked at many different factors and said which of these are explaining or shaping the microbiome of babies. And they looked at many things like probiotics and antibiotics that you would expect would change the microbiome, and they do. But what changes it much more, as you can see here, is breast milk. By far, the single biggest factor shaping the microbiome of babies. So again, I wanted to know why. And that's what I focused on when I started my own lab in 2014. And so to understand why, we have to think about what's in the breast milk. These are 2 pictures giving you the same message, which is that the ingredient list of breast milk is a whole lot longer than the ingredient list of formula. And in fact, this list is not even complete because we're still discovering new things in breast milk. And I know you can't read these tiny words, you can read these ones, and you can see that, of course, formula has all the nutrient building blocks a baby needs, but on top of that, breast milk has antibodies that I mentioned before, hormones, growth factors. And I'd say this figure needs an update because it doesn't contain 2 of my favorite things when it comes to the microbiome, which is probiotic or good beneficial bacteria and prebiotic oligosaccharides that feed those good bacteria. So these are naturally found in breast milk. And if you're thinking to yourself, but Megan, formulas these days have probiotics and prebiotics. It's true, but it is not the same. There's 1 or 2 probiotics, 1 or 2 prebiotics added to some brands of formula, but we know there are hundreds of probiotics and hundreds of different prebiotics in human milk. So in my team and in the field in general, we're starting to think of human milk as a system of all these many bioactive components. And in the past, it was studied in buckets. So the nutritionist would study the vitamins, and the endocrinologist would study the hormones. But the reality is these things are all mixed together in milk, and that's how the baby receives them. So that's probably how we should study them. We also recognize that human milk is a system embedded in another system, which is the mother-milk-infant triad. Many people talk about the mother-infant dyad, but we're recognizing that breast milk is an important and distinct 3rd component. And it's not just a one-way street. It's not that moms are giving milk to their babies and that's it. Babies are actually communicating back to their mothers through the act of breastfeeding, and that's super important. And then all of this is embedded within yet another system, the physical and social environment that can shape breast milk composition, that can influence a mother's ability or decision to breastfeed. And then this all together, of course, influences the development of many systems in the baby. So thinking of all these layers of systems and data is complex and requires big teams. You've heard that a lot today. My team is the child cohort study. So a lot of the research I've done has been embedded in this great Canadian study that was started back around 2009 when I was just getting into the field. It's a pregnancy cohort of over 3,500 families recruited from 4 provinces in Canada, including here in Toronto, as well as Manitoba, where I'm now based, Alberta, where I did my postdoc, and Vancouver. And so many researchers representing many disciplines are involved in the child study. It was founded to really answer the question of how genes and the environment influence child health and development. Very broadly, initially with a focus on asthma and allergies, which are so much more prevalent today than in previous generations. But as the study goes on, we start studying other outcomes. And these babies are actually now 12 years old, and we're still following them, which is pretty incredible. And so the research I showed you earlier on the microbiome and breastfeeding was part of the child study. So I've been part of the study for that long. And what I'll tell you about is some of our latest research from the child study on breastfeeding and the microbiome. This came out just last month, a really exciting paper where we took a step further from that initial research just showing that breastfeeding was important to the microbiome. We did that actually in 24 babies at the very beginning of the study. This latest research involves over 2,000 babies. And what we showed was that breastfeeding is really important for the microbiome, and that does have an influence on health. So a normal baby is going to gradually collect more microbes as it gets older and gets exposed to more things. And that gradual timing is really important. So we saw that babies who were breastfed had this slow and paced expansion of their microbiome. But babies who stopped breastfeeding too early had this premature acceleration of their microbiome development, and that was linked to a higher risk of asthma. We looked at this not only in the gut, but also in the nose of the babies in collaboration with our colleagues here in Toronto and a computational biologist in New York. So the message here is that timing is everything. A healthy microbiome is not just about having the right microbes. It's about having them arrive in the right order at the right time. And breast milk is really what drives this timing. So of course, we have more questions. How does that work? How is it that breast milk is driving the timing of the microbiome development? For that, we want to look at the components of breast milk. So here's an updated version of the figure I showed you earlier. It does have the pre and probiotics over there with formula. So my dream with the child study was to, one by one, analyze all these different building blocks of breast milk. And I did that pretty slowly at first, with pretty small grants. And a lot of this work is published. You're able to leverage that work and convince people that we need to look at all the blocks all together in a system. And so we were funded by the Gates Foundation to develop the International Milk Composition Consortium and really start analyzing that whole rainbow of milk. So the IMIC Consortium involves a collaboration with scientists and research participants here in Canada, but also in Burkina Faso, in Pakistan, and in Tanzania. And we're trying to understand how human milk is similar and how it's different in different parts of the world. And so we're doing that in over 1,000 mother-infant pairs, and we are looking at the whole rainbow of milk. We're also working with data scientists. When you generate that much data, you can't use traditional statistics. So we have experts in machine learning and AI working with us to unravel this complex tangle of data around human milk and to see how it relates to infant health and development in different parts of the world. And so this is where I'll say, come to my talk tomorrow to hear more, because we have limited time today, but I will share some preliminary results from the IMIC study, and I think you'll be amazed. So I'll switch gears a little to talk about MILC, the Manitoba Interdisciplinary Lactation Center. So this is a group that I founded in Manitoba to really think more broadly about milk. So we're establishing an infant feeding database where we hope to collect data on all infants in Manitoba to understand better how they're fed, because that isn't done well, and we can't fix something we don't measure. We're also starting a human milk biorepository, asking moms to donate milk for research to understand more about how it might predict their own health, maternal health, which is an exciting new area, and of course how it links to their infant's health. These data are all linked into our administrative databases in Manitoba to generate a really powerful and unique platform for milk research. And we have a milk club. This is where every month we meet between researchers and clinicians who care about breastfeeding to think about what what new research needs to be done and to share the latest results so they can get it out to the patients and practitioners that need to know about it. We meet every month, and we've invited, by popular demand, people from anywhere to join us. So it's not just Manitoba anymore, and if you're interested, I encourage you to reach out. Some of the things we do at Milk include initiatives to recognize that breastfeeding is a societal responsibility. It's not on individual moms to breastfeed. They need support. And so we're doing things like developing courses to teach healthcare practitioners about breastfeeding and lactation, because unfortunately, so far, that's not a mandatory part of their training. We're also generating videos for the general public to get them aware and excited about breast milk. We've generated policies for the Parliament, briefings to influence policies in Parliament. I'd love to generate policies. And we're doing events for the general public. So we had an AMA on Reddit for the first time this year. Super cool to see what the general public is wondering and asking about breastfeeding. We've established a milk mentorship program in Manitoba. So partners of ours in the Milk Club have established this program where a new mom who might be struggling with breastfeeding can get paired up with a mom with experience and get the help she needs. Maybe she doesn't need to go to a doctor or a community support where that is limited, but she can have a peer mentor leader through it. And two of our members of the Milk Club are founding members of the North American Board of Breastfeeding and Lactation Medicine. It might surprise you to know that this didn't exist until 2 years ago. There was no medical specialty in lactation. There's a medical specialty for every other part of your body, but not for the mammary gland. And so that's changing, which is really exciting. So in terms of what's next for our team and our group at Milk, I'm excited to do more milk multi-omics. So I've talked to you about the child study. We're doing this in other cohorts and also in the area of premature infants with collaborators actually here in Toronto, which I think is really exciting. We're studying colostrum. So colostrum, also known as the original liquid gold, is the very first milk that a mother produces in the first couple of days, and it is this golden color. It's because it's so different in composition. It's full of those antibodies and many other things. And so we've got new projects to study colostrum in different areas of the world, how it influences newborn health, and also looking at antenatal colostrum. So moms can actually start producing this before the baby is even born, and we're interested in the properties of that antenatal colostrum. We're also wanting to do more around breastfeeding support, so expanding that Milk Mentor program to rural and remote areas of the province, starting a pump loan program. So breast pumps, unfortunately, are not covered by insurance in Canada, something else we're aiming to change. But until then, we want to have a program so that any mom, whether or not she can afford a breast pump, can have access to one. And we want to do more in the area of knowledge translation. So we're working on a mini-series or a documentary about breast milk and breastfeeding. We're looking at ways to educate kids about breastfeeding early in their school careers. Now, I mentioned breast milk is a superfood for babies, but something pretty cool to think about is that maybe breast milk's benefits are not limited to babies. Of course, they should be first in line to get breast milk, but I think there's a lot we can learn from breast milk and translate to people of all ages. And so this is an interview I did recently with The Economist, and we talked about projects like this one. This isn't my work, but it's really exciting. So people who get a bone marrow transplant, they have to first wipe out their immune system so that they don't reject that transplant. Obviously, wiping out your immune system could have some side effects. And so someone had the clever idea that, babies don't have an immune system, but they get it from breast milk. So maybe we could give breast milk to these patients, or maybe parts of the breast milk to help them recover better. Another example of how breast milk could benefit everyone is irritable bowel syndrome. So this is a disruption of the gut microbiome. We know that breast milk is tailored to feed a healthy microbiome in babies, so maybe we could extract some of those components and help treat people with IBS in adulthood. So I think we have a lot to learn and be inspired about from breast milk. I want to switch gears a little and tell you a story of how I got here, a very brief one. Yesterday, I told the whole story, and I think that's recorded somewhere on YouTube, but I'll give you the highlights. Started as a patient, actually. So my parents are here in the audience today. When I was about 10 years old, I had breathing trouble when I was playing soccer, and they took me to the hospital. I got diagnosed with asthma and enrolled in a study. And that experience of being in a study inspired me to think, maybe that's what I'd like to do studies and help other people be healthy. So then a bunch of stuff happened. I went to school, and in 2014, I eventually started my own lab at the University of Manitoba, and it was just me. A few more things happened. I did some more school, got a few awards. In 2019, I founded Milk, and I got into CFAR, which was mentioned earlier. And then today, 2024, I'm here receiving this award, and my lab now has 14 people. But the point I want to make for you today, without going into all the details, is that that looks like a straight line, but actually, it was more like this. So sometimes you maybe feel like you're taking a step back, but that is totally normal and that is okay. So 2 examples for me are that I did my first degree in biochemistry. I thought that's what I wanted to do, and then I figured out along the way that maybe that wasn't my favorite, but I discovered epidemiology, population health, and I loved it. So it felt for me a bit like, did I waste 5 years doing this PhD? But looking back, I see that that wasn't a waste. You just heard what my research is about. It's about microbiomes and biomolecules in breast milk. So that totally makes use of my previous training, but also the new training. So what might feel like a step back isn't necessarily. Another example is when I finished my postdoc and I'd looked and studied the gut microbiome of babies, I thought, I'll keep doing that. But I was told, Megan, there are many more people in line ahead of you to get their hands on the stool samples from the child study. So I had to think for a moment, and I decided, well, I love this child study. Maybe there's something else I can do. And there were freezers full of breast milk that nobody wanted. So I took that on. But as you can see now, I'm linking it back to the gut microbiome. So again, what feels like a step back or a barrier might not turn out to be such a barrier after all. So there are many possible paths, not one right path. And it's okay to change your direction or your destination. This has also taught me the importance of mentorship, teams, and networking, because none of this research was done alone. I had lots of important mentors along the way, and you never just know what person in your network might be able to open the next door for you. The other thing that's hidden in this map is a lot of failures. So we talk a lot about our successes, our big papers, our big grants, but there's lots of failures for everyone on their way to success. And so everything I highlighted here in red is something that didn't go right the first time around. I had to apply a second time, sometimes a third and a fourth time before it worked out. And probably my favorite failure on this slide is my first grant on breastfeeding, where one of the anonymous reviewers made this comment, that it is highly unlikely that any magic component in breast milk will ever be found. Of course, I did not use the word magic because I'm a scientist, but that was the result. And early in your career, you can imagine that feels pretty disappointing. I thought maybe I should do something else, but you'll notice that was just 1 year before I saw Cesar Victoria win the Gairdner for breastfeeding research. So I kept at it, and I'm here today having this work recognized. So I think you don't give up just because one person tells you it's a bad idea, even if they feel like a really influential person. So failures are an inevitable and an essential part of success. I want to finish with one last quote. So, of course, we all know about the Nobel Prize. In that same Lancet series on breastfeeding, Keith Hansen, the Vice President for Human Development of the World Bank, said, "If breastfeeding did not already exist, someone who invented it today would deserve a dual Nobel Prize in medicine and economics." And so, of course, breastfeeding can't receive the Nobel Prize because it does already exist. But the Gairdner has now recognized 2 researchers for human milk research, breastfeeding research, so maybe that's not such a crazy prediction from Mr. Hansen. So I want to finish by thanking all the people who contributed to this work. Of course, my lab, members past and present, the child cohort study, the members of Milk, and the IMIC Consortium, all of our many funders, and because today is a special kind of day, I also want to thank the many, many colleagues, mentors, and family who have supported me to get here today. Some of them are in the audience, my parents, my partner. This doesn't happen alone. We need teams, we need mentors, we need family and support. So thank you. I appreciate you all being here, and here is where you can find me. We have no questions today, but you can reach out online, and I really appreciate the Gairdner Foundation for recognizing this award and providing this opportunity.
Great. Okay, so Dr. Megan Azad has selected our second 2024 Canada Gairdner Early Career Investigator, and that is Dr. Sue Tsai. Dr. Tsai is a Canada Research Chair in Immunometabolism and Diabetes and an Assistant Professor in the Department of Medical Microbiology and Immunology at the University of Alberta. She completed her PhD in Dr. Per Santamaria's lab, where her thesis focused on tolerance induction in type 1 diabetes. She then completed postdoctoral training with Dr. Dan Weiner here at the University Health Network in Toronto, and during that time studied mechanisms of immune dysregulation in models of obesity-related insulin resistance. Using a combination of animal models and immunological approaches, her lab studies the role of immune metabolic crosstalks in diabetes, infection, and cancer, and explores potential new strategies to improve immunity. Dr. Tsai will be presenting maternal humoral factors modulate offspring gut immune homeostasis to mitigate diabetes development. Please join me in welcoming her to the stage.
All right. Thank you for that kind introduction. It is a huge honor and also a humbling experience to share the stage with the laureates that pioneered the technology that we rely so heavily upon in my lab. And it's also inspiring to speak after Megan, who provided such important translational aspects of maternal factors and breastfeeding research, which is so relevant in so many chronic diseases, including the ones that my lab is passionate to study. So as Megan has beautifully explained, breastfeeding and early life exposure to maternal factors play a huge, profound, and long-lasting role on an individual's resilience to diseases through shaping their microbiome, through shaping their immune development, and this long-lasting effects persist long after the withdrawal of these initial early life maternal influences. So this is really an important field to study, and one good example of a disease that is altered by maternal influences is type 1 diabetes. We know that type 1 diabetes, or kids that are born from mothers with type 1 diabetes, they develop resistance to disease compared to kids that are born from mothers without type 1 diabetes, suggesting that the affected mothers are conferring protective factors to these babies. But to this day, we don't fully understand what are the mechanistic underpinnings of this protective mechanism that are provided by the moms. Type 1 diabetes, for those immunologists in the audience, is an autoimmune disease. It is mediated by autoreactive T cells, CD4 and CD8, that recognize peptides expressed or presented on MHC by antigen-presenting cells. And these are peptides that are expressed by beta cells that produce insulin in our bodies. So what happens then is that these T cells can home to the islets and mediate beta cell destruction, and that results in patients that become lifelong dependent on insulin injection to maintain their glucose homeostasis. This disease is shaped by genetic factors, but we also know that there is a heavy influence from the environments. And if we look at these environmental triggers or modifiers of disease development, you can see that many of them fall within this early life period, so during the prenatal or postnatal stages. Many of these have to do with infections, diets, mode of birth, nursing, and we know that all of these factors have one thing in common, and Megan had elaborated on this, which is the microbiome. So all these factors can contribute to shaping the microbiome in individuals. So in type 1 diabetes, we know that dysbiosis, or an imbalance in the microbiome in the gut, can impair intestinal barrier function, and through that allow bacterial products as well as metabolites to increase their translocation, gain access to the immune system, and aggravate autoimmune responses. But we also have built-in protective mechanisms, and one of them being this green dimeric antibody here. This is IgA, and we know that IgA can bind these bacteria in multiple ways to restrain them and protect against dysbiosis. So what IgA does and how it's linked to type 1 diabetes is not entirely clear. We know that in patients with type 1 diabetes, there's an increased incidence of what's called IgA deficiency. So this is characterized by low serum IgA, which is different from the mucosal dimeric IgA. But we also know that the fecal IgA, they bind bacteria in a way that's very different when we compare patients to healthy controls. We also know that IgA is the predominant immunoglobulins secreted into the gut as well as the breast milk. So this led us to ask the question, what is the role of maternal IgA in type 1 diabetes? To answer this question, we had to make an IgA knockout mouse. This was 8 years or 6 years ago when I was a postdoc, and I convinced my supervisor to let me breed mice in the facility here on the 6th floor. And we spent 2 years making this mouse. And then in my lab, when I started at the University of Alberta, we started following these mice for diabetes. But to answer this specific question, we had to do a very specific kind of animal breeding. So we had two breeding strategies. We generated heterozygote pups, so they are heterozygotes for IgA. But in one cohort, we had these pups that are born and nursed, or birthed and nursed by IgA sufficient dams, so they would have received maternal IgA for the first 21 days of their lives. And on the other hand, we have pups that are genetically identical to these pups, but they did not receive maternal IgA during this nursing period. But note that both pups are capable of making their own IgA. So we spent 32 weeks to follow diabetes incidence, because that's how this model works. And as you can see, these mice develop spontaneous type 1 diabetes because of their genetic predisposition. But after following all these mice, we proved ourselves wrong. We thought that maternal IgA deficiency would exacerbate disease. But to the contrary, we found that maternal IgA deficiency protected the mice against type 1 diabetes. We looked at the immune responses in these mice and the phenotype using tetramer staining and tracking for autoreactive T cells showed us that these mice are indeed, they developed diminished autoreactive T cell responses to islet antigens and were protected. So at this point, we were disappointed because we were a startup lab and we didn't know where to go. But then we kind of put our feet down, and then we decided we need to figure out what is protective that these dams are giving to the pups, because that could be beneficial in terms of thinking about the early life influences and how that modulate diabetes development. So we pressed on with our project, and we keep doing experiments to try and understand this relationship better. So one of the experiments that we did was we did a cross-fostering experiment where we swapped the little pups at day 1 of age onto either a wild-type IgA dam or a knockout or IgA knockout dam. And what we found was that just by being nursed by IgA deficient dams during this early life 21-day period, we could recapitulate the protection that we saw in the maternal IgA deficient pups. So we concluded then that maternal protective factors are transferred during nursing. So we know that during nursing, the dams can provide the pups with either milk or microbiota. We're following on the microbiota, but I want to share our data on the breast milk study because it was really a challenging effort to mechanically extract milk from mice. You can ask me about this later. But my student, she prevailed. She got enough milk so that we could do proteomic analysis of the 2 different milk samples, and we were able to identify more than 3,000 peptides, and a lot of them were very different or up or down regulated in our IgA knockout milk. And you can see highlighted in purple that many of these proteins that were altered were antibodies, so echoing to Megan's talk on how important that breast milk antibodies are in shaping the disease development in the pups. So we did then next look at the antigen specificity of these breast milk. We looked at what they were binding in terms of the fecal bacteria communities. And to make a long story short, what we found was that the milk from the IgA knockout dam, the antibodies within that milk are much better at binding specific strains of bacteria, and those strains of bacteria are suppressed in the corresponding pups, suggesting that there is an interaction between maternal milk and the gut microbiome of the pups that could be involved in diabetes protection. So we are still following on that to see if we actively induce immunity to these strains of bacteria, can we then protect the pups from developing diabetes? We also looked at how this altered microbiome and maternal breast milk composition altered or impacted the gut environment in the pups. We did RNA sequencing, and we saw an upregulation of pathways that are involved in antimicrobial resistance, defenses, pro-inflammatory pathways, and we validated these with real-time PCR. And so there's clearly more inflammation in the gut of our protected pups, and this was at 3 weeks of age, and it also went against a little bit what we predicted would happen. We looked at the immune composition in the gut tissues, and we saw that the gut T cells were more activated. We saw an expansion of IgA plasma cells, and importantly, we also saw an expansion of induced regulatory T cells in the gut. And when we compared the mucosal barrier of our protected versus non-protected pups, we saw an increased thickness, a more robust mucin layer in our protected pups compared to the ones that were not protected. And so this is what we think is happening, although a lot more work needs to be done, but we think that the maternal IgA deficiency alters the microbiota, which interacts with maternal humoral factors to induce a strong inflammatory event during weaning. This is what's called a weaning reaction, and that leads to beneficial imprinting of the gut, bolstering the barrier function, expansion of regulatory cell types that then result in the suppression of autoimmune diabetes in the islets. So I want to end by echoing back again to Megan's talk to say that timing is really everything, and we still have a lot to do to understand the maternal factors and how they influence the development of type 1 diabetes. But we really truly believe that this early life period is crucial and is a window of opportunity to develop prophylactic strategies to curb this disease. So I would like to thank my lab. Erin was the one that developed the milking and most of the work in this talk, and also my other trainees, my collaborators, and also my mentors and funding agencies, because without them, this work would not be possible. And thank you.
Our second Momentum awardee is Dr. Christian Landry, Professor of Biology and Biochemistry at Laval University and the Canada Research Chair in Cellular Systems and Synthetic Biology. Dr. Landry completed his Bachelor and Master's in Biology at the Université Laval before obtaining his PhD from Harvard University and completing postdoctoral training in Systems Biology and Biochemistry at Université de Montréal. Dr. Landry started his independent position as a CIHR new investigator and a Fonds de Recherche de Québec Santé junior investigator in 2009. In 2015, he was appointed Canada Research Chair Tier 2 in Evolutionary Cell and Systems Biology, and in 2020, appointed Canada Research Chair in Cellular Systems and Synthetic Biology Tier 1. Currently, Dr. Landry is also leading a pan-Canadian training program on the evolution of fungal pathogens funded by NSERC. Dr. Landry recently received the Université Laval Excellence Awards in Teaching and Mentoring, the SMBE Margaret Dayhoff Mid-Career Award for Outstanding Contribution to Research, and the NSERC EWR Stacey Memorial Fellowship, and in 2014 was elected to the Royal Society of Canada's College of New Scholars, Artists, and Scientists. The 2024 Canada Gairdner Momentum awardee, Dr. Christian Landry, will be presenting his presentation, the title changed here, How Evolutionary Biology Brought Me to Biomedical Research. Please welcome.
Thank you. All right. Thank you very much for having me. It's super intimidating presenting in front of so many great scientists. Also intimidating to be presenting in front of my son and my sister, I think, who are seeing me for the first time talking about science. I want to start with telling you about my journey that brought me here, because it's a little out of the ordinary. So I started in biology, and I thought when I started biology I would study big mammals. I would become a naturalist in the Gaspésie National Park, where I come from. And as an undergrad, I studied developmental biology in a cancer lab. And as a master's student, I discovered that I was interested in biology. I studied the population genetics of salmon, a species that we like in Canada. And then as a PhD student, I was still interested in evolutionary biology, and I joined Harvard University to study the evolution of sea urchins, how sea urchins become, one species become two species. And something really major happened in my life. My PhD advisor left Harvard, he went to Stanford. I didn't go with him, so I had to join a new lab. And that's why I joined the lab where they studied molecular evolution using fruit flies and budding yeast. And I started studying evolution, but at the molecular level. And that's what I've been doing ever since. And when I came back to Canada, I really wanted to know more about proteins and the way that cells work. So I joined a lab in biochemistry where I studied protein-protein interactions. And that's what made me who I am now. And everything that I've been studying in biology is under the lens of evolutionary biology, because that's the thing that I discovered in biology that made biology a little bit like physics and mathematics, where there were principles and things that explain how the diversity evolved in nature. There were main forces that we could study, and that's what made biology interesting to me. And so biologists have been studying evolution for a long time. And the main thing that we know about evolution is that thing that Darwin put together is natural selection. And we all agree this is the major engine of evolution. But if natural selection is the engine, what is the fuel of evolution are actually mutations. That's what can feed into evolution. And evolution is the algorithm, and mutations are the data. And thanks to the development of technologies that we've heard of today, we can now study mutations systematically. So surprisingly enough, the thing that we didn't know a lot about up to recently is what mutations do and how do they change the biology that makes things improving with time. So now we can use synthetic biology to create mutations, and we can use high-throughput sequencing to read mutations, and we can now associate the impact of mutations with what they are, and that changes the way that we do biology now. So the context in which we study that is in the context of antimicrobial resistance. So one of the major progresses we've seen in human history is the increase in life expectancy in humans. So it has increased more than twofold over a few hundred years. And one of the reasons why this is happening, and that's actually the main reasons why this is happening, is that we can control microbes. Many of the things you've done since this morning is actually controlling microbes. You wash your hands, you wash your teeth. All of that is contributing to making us not die when we're young. But what happens when we do that is now that we become the agent of selection. We're selecting microbes, and we're applying selective pressures on microbes, and microbes change in response to that. And of course, the context in which this is the most important right now is in the context of antimicrobial resistance. So, there are many numbers on antimicrobial resistance. I can just tell you one. People estimate that now there may be 1 million people dying every year because antimicrobials don't work. That number is going to increase by tenfold over the next 25 years. More people need antimicrobials. We have more people with weakened immune system, and we're simply more people on Earth. So we need to do something about it. And so this is what brought me to applying evolution to biomedical research. And we study something that's a bit different from what many people study. There's a lot of problems with antimicrobial resistance in bacteria. We study fungi. So I was trained on working with the budding yeast, the model system in cell biology. And many of the fungi that infect humans are super close to yeast, so we can use the tools that we know of and contribute to that. So fungi don't infect humans very often. They usually are a problem with people with weakened immune system, but we carry them all the time. If our immune system is weakened, they can invade some organs, and they become an important problem. And in spite of their importance, and that we realize now, there's a tiny fraction of all funding dedicated to pathogens that's actually dedicated to studying fungi. So it's a good timing to be involved in that type of research. So the way we see antimicrobial resistance in fungi is we see that as an evolutionary problem. So you can imagine when there's an infection, there's a population of fungus, yeast, for instance, that decreases in size. And then we detect it, we start treating with drugs, and we hope that there won't be any mutations that happen before we can decrease the population. So it means that what's happening in a treatment is a race between mutations and the impact of treatment. And if there's a mutation, resistance mutation that occurs, then treatment is going to fail. So we set up a research program where we want to measure like this aspect of antimicrobial resistance, which is what these mutations that can cause resistance. And we're trying to understand why can it evolve so fast. So for that, we think we need to measure how many mutations in a genome can confer resistance. Because the more mutations there are in a genome that can cause resistance, the easier it is or the faster a mutation will occur that causes resistance. And then we want to know if there is resistance to one drug, is that mutation leading to resistance to many other drugs? Because it's going to change the way we treat patients. And finally, one important question in the field is, are mutations causing harms to the pathogen? Because if a mutation decreases the fitness of the pathogen, when we stop treatment, that mutation is going to disappear. It's going to be selected against. If it doesn't cause harms, then it might stay in the population and could even be transmitted. So that's a parameter that's super important. So if I summarize these things that we want to measure, so you imagine there's a mutation, a resistance mutation happens in a single cell first, and then you apply selection with a drug, the frequency of that mutation increases, and then then you discover that a treatment doesn't work, you might stop it. If there's a cost to that mutation, so if that mutation decreases fitness of the fungus, it's going to disappear. If there's no cost, that mutation can stay in the patient or it can even be transmitted. And then the other important factor is now you can change treatment. You can choose another drug. But if that same mutation causes resistance to many drugs, there are not many options you have. In fact, you might not know, but you might not have any options if a mutation causes resistance to all of the drugs that are available. So the way we want to quantify the impact of these mutations is to do that systematically in the genes that are known to cause resistance. So what we do is that we take a gene or a protein of interest that is involved in antimicrobial resistance, and we make libraries of mutants in those genes, and we measure the impact of all these mutations on resistance to the drugs of interest. So we've been doing that for the past few years, and there are like 4 classes of drugs that are used to treat fungal infections. 3 of them, they specifically target proteins. These proteins here that we've been studying. So we've been doing these kinds of experiments I will tell you about in a few minutes. So I'm going to tell you just about one story that we just published, but the idea is the same for all these studies that we recently started doing. So what we do is that we create diversity. We take that gene of interest, we create all the possible single amino acid mutant in that protein, and then using yeast as a model system, the Baker's yeast that we know of how to work with in the lab, we can combine all of these mutants in a single flask. We grow them, we can compete them with drugs or without drugs. And then we use, that's where high-throughput sequencing becomes useful. We can follow the frequency of all these mutants in the competing populations using high-throughput sequencing. And then we know we can track all of these mutants in the tube, and we can see if we're growing that population in the presence of drugs. We can see that mutants that cause resistance are the ones that increase in frequency. They are positively selected. And the ones that decrease in frequency are the ones that actually perturb the function of the protein without conferring any advantage. So we can do that with proteins like this one. This one is the target of a class of drug that's called azoles. So that's the most frequently used drug to treat fungal infections. And it is an enzyme that processes this molecule here, and there's an active site in the middle. And what azoles do is that they block this active sites. And resistance mutations, they can prevent the drug from coming inside the protein, and that's how they confer resistance. So what we can do is that we can take a gene from a fungal pathogen, express it in our model system, In this case, we take this particular protein called ERG11, and we mutated all the sites around the active site of the protein, which is basically where the drug usually enters the protein. And then we can express the gene from that fungal pathogen in the model system, and we use tools of synthetic biology to just repress the gene that exists in that species that codes for the same protein. So the cell here is going to grow, and all growth will depend on the gene from the fungal pathogen. So if we want to answer the question of cross-resistance among molecules, we can do the experiments I just told you with a bunch of drugs. There are many azoles that are used to treat humans that are infected with different fungi. So we can do the kind of experiments I told you about with all of these drugs and measure the contributions of mutations to resistance and see if there is cross-resistance. So the kind of raw data that we get is going to be shown on this screen. It's basically a dictionary of mutations, all the possible mutations in that protein. So we have here on the x-axis all the positions that we mutated in the protein. On the y-axis, all of the possible amino acids that were inserted at those positions. And then the color represents the impact of those mutations. So this experiment was done in the presence of azole. When it's green, it means that these particular mutations, they confer resistance to the drug. And brownish red means that these mutations are harmful for the protein. They decrease the activity of the enzyme, but they don't confer resistance. So we can create these kinds of maps for the set of positions that we studied in this particular protein. And then you can see there are hotspots in the protein that confer resistance. Those are presumably mutations that prevent the drug from entering, but that maintain the function of the protein because the protein needs to be functional for the fungus to be resistant. So this is the kind of experiments we do. Now we can ask, so this is a map for one drug. If we do that for all the other drugs, what kind of images do we get? We actually, surprisingly, although the molecules look different, we surprisingly find exactly the same kind of image. So we did the experiment with many drugs, and with that we can show that there's a lot of cross-resistance. What that means is that if there's a mutation in a patient that prevents a drug from working, 70% of the time, all of these other drugs will fail treatment. So this is something super important to be measured. And also, it shows how important it would be to have this data when you switch treatment from one drug to another. Now, the second thing that I mentioned is super important to measure is the cost of these mutations. So we can measure how these mutations impact the normal function of the protein, which I am representing on the y-axis here, which is a measure of the impact of a mutation on the function. Negative means negative impact. And we can measure the impact of the same mutation on resistance. And what you can see from this plot is the following. So you see every data point, every point is the impact of one mutation. You can see all these mutations, they increase susceptibility to the drug, so they are interesting mutations. And only a tiny fraction of the mutations, they lead to resistance and are deleterious for the protein. And in fact, most of resistance mutations, 90% and more, they increase resistance, but they cause no problem to the protein, which means that these mutations, if you stop treatment, they won't disappear. They can stick around because they don't impact the fungus that much, or not at all. So this is a kind of tools that where we combine the kinds of experiments where we combine synthetic biology and high-throughput sequencing to produce data that's useful from a point of view of evolution of resistance, but also I will tell you how we can use that data practically in terms of medical application. So for the last few minutes, I want to tell you a few things that I also did in the past few years. When I got into that field, we realized that there was very little training among people who study fungal pathogens in terms of evolution. Although I told you this is a problem, an evolutionary problem, if we don't understand evolution, I think we cannot deal with that problem perfectly well. So what we did was to create this training program across Canada that started 3 years ago with many different universities. So I led the creation of this program through NSERC Create training grant. And we're about 10 PIs across Canada who work on fungal pathogens of humans, but also of plants, because there are many things that are shared between plants and human pathogens. And for the past 3 years, we've been training PhD students and postdoctoral fellows across Canada. They work together, they take classes together, we created new classes, and we train them on the evolution of fungal pathogens, also on technologies like CRISPR-Cas9, genome editing, high-throughput sequencing, and bioinformatics, so they can implement now the kind of experiments that I told you about. So far, we've trained nearly 60 fellows, and we only have 2 in the program. So I think we're contributing to the next generation of people who can help solve the problem of fungal infections. So one of the concrete and cool thing that we did with this group of people in the past few years was that we realized when we were looking for mutations that all the data that people had collected over the years on resistance mutations, they were scattered in different papers, like papers reporting a single mutations. I showed you the experiments where we can track like thousands of mutations at the same time, but we thought that mutations coming from clinical isolates were super important to report and be put together. So we took I invited many of the fellows in the program, and I said, let's spend a few weeks together. We're going to read all of these papers and collate all of the data so we can create a database that now people can use in the field, and they can go to the database instead of trying to find mutations in the literature. So we curated many papers, several hundred papers. We ended up using data from 400, a bit more than 400 papers. And now we have like thousands of associations between mutations and resistance to antifungals. So what can we do with that? So when I received the instructions for this talk, they asked me, what's the impact of your work? Most of what we do is super fundamental. That's why I chose to talk about this particular project. But here there's something super useful that we can do, and we're actually doing with public health labs in Canada. And how you can use this information is to try to assess more rapidly the susceptibility of fungal infections to antifungal treatment. So the way that clinicians do in Canada, at least, when they have a patient who's infected with a fungus, is that they take a sample, they grow the sample, they hope it grows, and then they isolate a few colonies of that fungus growing. They send it to public health labs, and in the public health labs, they grow them and they do the phenotypic testing. They say this drug works, this one doesn't work. They call back the doctor and they tell them, you know, you should be using this drug rather than this drug. Sometimes they just start with the standard procedures and they might adapt depending on the results. This works perfectly well, but it's sometimes too slow for some patients who are critically ill. What we're trying to do with the public health labs is to try to skip all of that growing step, and we try to work with them so we can take DNA samples directly from the patients, sequence the genome of the fungi, and then use AI-based technologies with the data that we collected so we can predict what the fungus is going to be resistant to. And with that, we hope that we can save some time. So that should take maybe 2 or 3 days, whereas this can take 2 or 3 weeks sometimes. So this is all the data that we collect can be useful in terms of medical application. Okay, just to summarize what I told you about, we are a super strong selective pressure on microbes, and microbes evolve in response. It's an interesting problem for an evolutionary biologist, but it's going to be a concrete problem for us in the future. And this is particularly true for antimicrobial resistance. And we think that by combining what we know about evolution, we can solve some of these problems more easily. And now we can make use of the high-throughput technologies, some of them that were invented by people in the room, which is quite amazing to be presenting that to you. And we can study these mutations before they even occur. So many of these mutations, they never occurred in the past or were never seen. So it's like accelerating evolution in many dimensions at the same time. So we can foresee what can happen, and then we can act better because of that. And what is going to come from that, I hope, is that one of the major problems when we study human fungal pathogens is that it might sound surprising for some of you, but we're super close to fungi in terms of evolution. Fungal cells and human cells, compared to all the diversity that exists, are basically the same cells. So it's super difficult to find proteins that we can attack with drugs that would kill fungi, but that would not kill humans. So we already have a set of good candidates for drugs, But the problem is that there are resistance mutations. So maybe what we can do is to modify the drugs to overcome resistance mutations rather than trying to find new targets and new molecules that are completely different. Okay, so that's it. That's what I wanted to tell you. I want to thank the people who do the work. Everything we do, all the stuff that I showed you is done and led by graduate students. And the funds, importantly, the funds from granting agencies. And then there are two important people on this picture who are also in the room, Isabel and Alex here. They are research professionals who've been working with me almost since the beginning of my lab, for Alex and Isabel since the beginning. And we work on all projects together. And I've been able to have them in my group because of sustained funding from CIHR, for instance. So that shows that funding is super important. We can keep these people around us, and they help make great progress in science. Thank you very much.
Okay, Dr. Landry has selected our next 2024 Canada Gairdner Early Career Investigator, who is Dr. Raquel Coelho-Martin. Dr. Martin joined the Department of Human Genetics at McGill University as an assistant professor in 2022. She undertook her PhD studies at Dr. Chapman's lab at the Wellcome Center for Human Genetics at the University of Oxford, and during that time described the mechanistic role of the DNA repair protein 53BP1 in optimal p53 tumor suppressant responses. Her postdoctoral work as an EMBO long-term fellow at Columbia University used CRISPR-dependent base editing to perform genetic screens at nucleotide resolution and functionalize DNA variants at scale. And this approach allowed her to identify loss, gain, and separation of function mutations and new functional domains in DNA damage response proteins, and to functionalize variants of uncertain significance in cancer predisposition syndromes. Now her lab applies cutting-edge, large-scale precision genome editing to approach complex biological questions and to improve intelligent drug design. Dr. Coelho-Martin will be presenting Understanding Unique Protein Features with Precision Genome Engineering. Please join me in welcoming her to the stage.
Well, first, thank you, everyone. Thank you to the Gairdner Foundation for this honor, of course, to Dr. Landry. And I also want to extend my thank you to the whole Canadian community, because as a newcomer, I have been welcomed here with open arms. So basically, that introduction said a bit of everything that I'm going to be talking about today. So let's just go into it. So we have heard today a lot about basic science and really fundamental questions, so I'm going to give an example of that during my talk. I have been working on this protein that you see up there, 53BP1, for almost 10 years now, since the beginning of my PhD, so I'm one of those researchers that worked on the same protein for their whole career. And I find it a very interesting protein. So you see it's fairly large. It has a very long N-terminal that is not structured, so it makes it tricky to study, but uses that as a platform to recruit factors to DNA damage sites. And it does so by forming big oligomers and recognizing two key histone marks as sites of lesions that are H4K20 dimethyl with the tandem Tudor domain and H2AK15 ubiquitination with the ubiquitin-dependent recruitment domain. And all this architecture is essential for the repair of DNA double-strand breaks by a non-homologous end joining. And this process is very important in physiological conditions. We have heard today about the generation of the T-cell receptors, so very important for VDJ recombination, also very important for the generation of the antibody repertoire. But it's also an extremely toxic pathway when we find BRCA1 deficient tumors. This non-homologous end joining drives the generation, as you see there, of these massive radial chromosomes and massive chromosomal rearrangements. And 53BP1 had been studied in this context for a few decades, but we were interested, we were interested on this extreme C-terminal region of this protein that is called the tandem BRCT domain. And this interacts with master tumor suppressor P53, and also with the deubiquitinated enzyme USP28, brings them together, and that function is essential for optimal P53 antitumorigenesis in response to different lesions, such as genomic instability, but also other groups reported that this is critical when you have extended mitotic division, and your cells could probably missegregate your DNA, and basically this pathway goes and kills those potential cells that will cause problems. So when I was studying this during my PhD, we are molecular biologists. We were using traditional molecular biology techniques. So first, you establish a loss of function model. So we used CRISPR-Cas9. That was just, I guess, just published back in the day to generate genetic knockouts for 53BP1 and also for P53. And we put a drug that is a specific P53 activator. The drug is called NATLIN3. So obviously, when you see there, when you don't have the target, you don't have P53, the cells are fully resistant. And when we didn't have our protein of interest, the cells had an intermediate phenotype. They were also resistant, indicating us that there was a functional relation there. And then to disentangle the mechanisms, because this is what we do in the lab, we are very interested in disentangling the mechanism of how things work. We will complement that phenotype, put back a wild type, 53BP1, and see whether we were able to revert that resistance, but also put back design mutants that we made based on how we thought the protein worked, right? So if you have there, what I said, the tandem Tudor domain, that's a methyl binding module, we will mutate the methyl binding pocket. And then see whether that was involved or not in driving that resistance, and then if it was doing so by fostering the formation of the complex with P53 and USP28 that are, the discovered binding partners. But this had a fundamental problem, right? All our structure function that we call it base analysis were biased, were not really systematic. It was based on how we thought the domains were, how we thought the protein worked. And also we had additional problems that prevent us from using demutational scanning and other more systematic approaches, because our proteins were difficult to express ectopically. You see 53BP1 is not a small protein. They were large, highly structured, heavily regulated. So this complex was really telling us, you need to figure something out to study this on the physiological context and in a systematic manner. So then I moved on to my PhD, from my PhD to my postdoc work, and that's what I set out to do. And we devised the implementation of CRISPR-dependent base editing at high throughput. And this is a simple schematic of how one of those experiments look like. So basically, we have our pool of cells that express a base editor. And in the case of the screens I'm showing here, this is a cytidine base editor. So we have the Cas9, a nickase format, fused to a cytidine deaminase that makes C2T transitions in a specific area of the genome where we design our guide RNAs to obviously bring that base editor. And then we bring that base editor to our genes of interest by putting guide RNAs all across the coding region of that gene and making those C2T transitions in a systematic manner. And after that, we obviously put those guide RNAs into the cells. We added P53 activating drugs to enhance those phenotypes. And then using next generation sequencing, we can sequence the end population versus the initial population and figure out which mutations are behaving like loss of function, giving us resistance to the drugs, or potential even gain of function, giving us sensitivity to the drug. So if we zoom in one of those maps that we have over here, you see actually that the mutations, we have nonsenses and splice variants in yellow and red as reference for loss of function. So you can see the mutations cluster in specific regions of the protein. They are not stochastically all across. And we were particularly interested on the mutations place in the tandem Tudor and the tandem BRCT domain because we had been studying those domains for quite a while and we thought we had it figured out by now. So those are crystal structures from the tandem BRCT domain. On one side, you can see the results from one of your screens. So in the purple, positive log-fold changes. So cells that were given mutations that were given resistance, loss of function. And then in blue, you have sensitivity. And then side by side, we have amino acid conservation. And what was surprising to us, because we usually attribute to amino acid conservation if a region is conserved. We think that that's functionally relevant or structurally relevant. And when we were seeing where our mutations were localizing, if you look for these dark spots over there, they were not really correlating with areas that were strongly conserved. Right down here, that was not really strongly conserved. But our mutational analysis was really telling us there was something important right there. And the same was the case for the 53BP1 tandem tutor. You have here in the front the methyl peptide binding site. That's the canonical way these domains bind, and it's very strongly conserved. But then all our mutations that we were finding here were mapping right there to the rear interface. And then we had some gain-of-function potassium gain-of-function mutations on the other side, but really not in an area of a strong evolutionary conservation. So we were quite puzzled by those results, and we decided to figure it out molecularly. That's what we do best. So we selected the specific mutations that were being made in the tandem tutor domain and validated it one by one in what we call two-color competitive growth assays. So basically, we put cells that are wild type, color here in red, cells with our mutation, color in green, and then we mix them and let them fight, and then see which one overpopulates the other. And basically, this told us, the ones that look like loss of function in our screen are having the exact same phenotype in these one-on-one assays, and the same with the one that look like a gain of function. And then we do deep sequencing at endogenous locus to verify exactly which are the mutations that are being inserted. And in this case, we had valine 144 to isoleucine, glycine 1562 lysine, giving drug resistance, so really loss of function, and G1593 to lysine, giving drug sensitivity. So we always go back to the structure, and you see they are placed right there, where I told you that dark spot was coming up in the structure before, and it's really on the opposite side of the methyl binding module. And so we were thinking, well, this is probably really not required for 53BP1 to go to sites of DNA damage, because unless it's allosterically regulating that, like it's so far apart. And that was really the case. So despite the presence of those mutations, 53BP1 was able to be recruited to double-strand break lesions like a wild-type control. We have the AABS1. And it was actually functional in those sites because it was enabled to restore homologous recombination in BRCA1 deficient cell as deleterious mutations will do. But again, once more, those mutations were critical in p53-dependent responses. We put them in NALIN3. Loss of function mutations were resistant. Gain of function mutation was sensitive, indicating that they were regulating p53. And this was perfectly correlating with their ability to form the complex between these three proteins. So both loss of function mutations had trouble binding to USP28, and then the gain of function mutation was actually binding better, so forming probably a better complex and regulating p53 better. So at this stage, we were very happy, and we were like, okay, great, we have figured some more things out. These type of screens, this type of systematic mutagenesis analysis allow us to discover loss of function, gain of function, separation of function mutations, and very interestingly, a novel protein-protein interaction surface that is not the way these domains usually interact, and is like super rare, and we have figured out how this protein binds to USP28. Right. Well, that maybe was not really the case, because we had all this knowledge, and when you have that knowledge, sometimes you go to the structural prediction softwares, and this was done with AlphaFold 3 multimer, and we're like, okay, now we can probably tell which prediction seems accurate based on our mutations, right? And this is the tandem tutor domain, now placed in a little bit of a different position. Right over there is the methyl binding pocket. Right over here, our pocket of interest, and we place USP28 there. And it was really not binding where we thought it will bind, right? Like our QIP was telling us that that mutation was disrupting, but it was really not binding there. And in fact, what was binding there was the tandem BRCT from 53BP1. And it was doing so with that region where we also localized mutations based on our analysis. I was like, okay, well, that was not at all what we were thinking. But if we put P53 right there in the mixture as well, you see that actually where we find mutations with our screens is where those partners are binding. You see there the P53 BRCT interface, and you also see there USP28 having contacts with the BRCT right where our mutations were identified from the screen. So probably, what we were seeing in our biochemical assays were just like the loss of this internal interaction was releasing the whole complex and moving USP28 out because both domains in 53VV1 are required to retain the interaction. So not really how we thought they worked in the first place. And of course, we do think these type of tools have a lot of potential for discovery, right? And we are not only interested on those questions, we are interested in most translational questions, so I choose a basic one, but we are also interested in others, such as how to use these systematic mutagenesis to map allosteric sites and find a way to track them, to identify novel functional domains, even in proteins that we have been studying for many, many years, as it happened here. And also we have some projects on using CRISPR-based technologies to identify targets for rare disorders. And this is, well, maybe what's going on in the lab, but then we have managed to convince different groups to work with us with these, and we have projects in collaboration ranging from regulation of mRNA transduction by mutating non-coding regions to understanding, maybe appealing more to this audience, TCR antigen interactions. And with that, I would like to finish by saying thank you to my lab members and also to my PhD and my postdoc lab that I saw some working here, and to all our collaborators. And also, I would like to also say thank you to all the labs that we have managed to convince so far to join our base editing screening crew, and they believe that this tool is useful for their research as well. And I think that that's the beauty of it, like when you have a tool that you think is useful for research across disease areas, share it, encourage the usage, and try to see to do that research all together. Thank you. Thank you, everyone.
Okay, the 2024 John Dirks Canada Gairdner Global Health Award awardee is Dr. Cherry Kang. Dr. Kang received her training in medicine and microbiology at the Christian Medical College, Vellore in southern India. She is professor of microbiology at CMC, where her research group has established substantial community-based birth cohort studies addressing childhood enteric infections, growth and development over 20 years. Using data and insights from these studies alongside immunological, mechanistic, epidemiological, and implementation research, Professor Kang has generated extensive information on the complex interactions between the environment and infections in children, and the influence of prior infections on subsequent responses to vaccination. As a physician scientist, Professor Kang has led studies that contributed to the development and introduction of 2 Indian rotavirus vaccines into the National Immunization Program. These vaccines are now WHO pre-qualified, and their introduction has begun in countries outside India. She has expanded her research and collaborations to work on other enteric pathogens, particularly cholera and typhoid, with a view to further vaccine introductions, on nutritional and pharmacological interventions aimed at improving oral vaccine efficacy, and on the transmission and impact of vaccine effectiveness for COVID-19 in the Indian context. Recently, Dr. Kang has also joined the Bill and Melinda Gates Foundation as Director, Enterics Diagnostics, Genomics, and Epidemiology. The 2024 John Dirks Canada Gairdner Global Health Awardee, Dr. Cherry Kang, will present her presentation, India for the World, the Story of Rotavirus Vaccines. Please welcome Dr. Kang.
Thank you. I know it's been a while, so can I just ask if everybody can just stand up and turn around a couple of times in honor of the rotavirus, wheel-shaped virus, right? Okay. Right. So thank you very much. I'd particularly like to thank the Gairdner Foundation for recognizing work in enteric infections and work that has been done in India. I have a problem recruiting people to my group because, quite honestly, to get people to work in an area that labels itself as trying to solve problems of children, and particularly diarrhea, does not appeal to stellar scientists around the world. So I think the one thing that I leave behind as a contribution is the fact that we actually now have people that are interested in the space and have begun to work on enteric infections and their consequences. So I'm going to be walking you through one part of my journey, which is the work on rotavirus, and particularly the Indian rotavirus story. Rotavirus is known to occur in both humans and animals. The human story starts in Australia, where Ruth Bishop looked down an electron microscope and saw that there were wheel-shaped particles in biopsies of children that had intractable diarrhea. She was able to identify those particles. They were originally called duovirus because people thought that they had a double-layered shell. We now know that it is a triple-layered shell, and rotaviruses are the commonest cause of acute gastroenteritis in humans, in cows, in horses, in birds. If you can find an animal and look for it when it is young, chances are, if it has diarrhea, it'll be a rotavirus. So rotaviruses are common in every part of the world. We call the rotavirus a democratic virus because it affects the entire population. The problem with rotavirus and with diarrhea in general is that if you go around the world and you talk about diarrhea, people either don't recognize that it is a problem, or they think it is really easily treatable because of oral rehydration solution. The problem, of course, is that viral gastroenteritis is different from bacterial gastroenteritis, which is what oral rehydration solution was made for, because in viral gastroenteritis, you get the diarrhea, but you also get vomiting. So when you have vomiting, it becomes very hard to rehydrate a child. So if you're leaking fluid at both ends and you have a large surface area, which is what happens to babies, they can dehydrate and die very quickly if you can't get to care. Sometimes if you get to care really late, it can be very hard to find an IV in a child. So people put IVs into the scalp, into bones in the leg, all sorts of efforts to try and get fluid into children, which are not always successful. So it's, as I said, rotavirus is a democratic virus. It affects everybody in the world, and hygiene delays, but does not prevent infection. And this is a reason when rotavirus vaccines were first introduced, the first country that they were introduced was in the US. It is a virus that, where IV fluids can help solve the problem. And as I said, if you can't get to IV fluids, children will die. So lots of people talk about diarrhea and say, well, why not water and sanitation? And water and sanitation do help. But if I was to rank order pathogens for which water and sanitation are most effective, it is bacteria first, parasites second, and then viruses. Now, rotavirus vaccines have been in development from the 1970s onwards. There were a lot of different approaches that were taken to development of vaccines. We followed the Jennerian principle, which is to take an animal virus, put it into humans in the hope that it would prevent a human disease. But the first vaccine that was licensed in 1998. It was the Rotoshield vaccine made by Wyeth, and Al Kapikian, who is shown here, was a long-term scientist at the NIH who developed this vaccine. This vaccine was really interesting because it had a human virus backbone. It had a monkey virus backbone, and on the surface of it, there were genes that had been taken from human viruses and put on top, because at that time, we thought that the diversity of rotaviruses would require a multivalent vaccine, much like influenza. Rotavirus, like flu, has a segmented genome. So before rotaviruses were introduced, this was the pattern of diarrheal diseases around the world. This is 1998, before vaccines were introduced. Now, does it make sense to introduce the vaccine into the US first? Well, it happened. The vaccine had been evaluated in about 10,000 children in clinical trials. It was rolled out very rapidly. It was given to American children. After about 1.5 million doses of these vaccines were given out, they identified an association between the vaccine and a condition called intussusception, which is a telescoping of one part of the gut into another part. When this was identified, the vaccine was withdrawn in early 1999, and subsequently trials were all designed to be large enough to look at the effect of intussusception. So if you look at the figures that are shown on top on the left-hand side, what you see there is that when you have the intussusception in association with the vaccine, it usually occurs after the first dose of vaccine and in the first week after the first dose. So a defined period and a defined event, which actually makes it reasonably easy to identify that this event is associated with vaccination. Now, at the time that the vaccine was introduced and then withdrawn, there were a lot of discussions around the world because rotavirus was killing about 500,000 children annually around the world. Most of the deaths, as I showed you, were occurring in Asia and in Africa. So people said, if this is an identifiable event, occurs within a defined time frame, why don't we go ahead and use the vaccine in any case? Because we will save many more lives than will be lost due to intussusception. This was discussed at the NIH, it was discussed at WHO, and then it came to the countries that were supposed to implement this vaccine, and the countries said, there is absolutely no chance that if there is a vaccine that has been rejected for American children, that we will use it in our country. So that led to new vaccines being developed, new trials needing to be done. Instead of 10,000 children, as was done for Rotoshield, now the trials had 60,000 to 70,000 infants that were enrolled. The trials were completed, and the two vaccines made by GlaxoSmithKline and Merck were both licensed in 2006. The vaccines, when they were introduced, were 3-dose and 2-dose vaccines. Didn't matter which one you got. It cost $200 to vaccinate a child in the US. It actually costs a bit more than that in the US now. So if these vaccines were to be introduced in India, I did a back-of-the-envelope calculation at the time, it meant that we would use about 70% of our entire government health budget on one vaccine for our population. So obviously, that wasn't a vaccine that was going to work for us. Nonetheless, these two vaccines came into the private sector, and they came in at something that was only affordable for people who could afford private health care. These were effective vaccines. 85% and 95% efficacy. They were different from each other. One was a monovalent vaccine based on a human strain. The other was a bovine human reassortant, unlike the rhesus human reassortant that had been withdrawn. So moving now to the Indian vaccine story, we had been doing surveillance in India through a network that was sponsored by the Indian Council for Medical Research. Shown here are all the sites where we were conducting these studies, looking to see what proportion of hospitalized gastroenteritis was due to rotavirus. And essentially, 2 out of every 5 children that were hospitalized in India had rotavirus. We did those hospital-based studies, we did community-based studies, and this is what we wound up calculating as a burden of disease for children in India. So in 2011, the birth cohort was about 27 million children. And we looked at what would happen to them over a period of 2 years. And essentially, 1 in 2 children would develop a rotavirus infection. 1 in 8 would have diarrhea that was severe enough to take them to outpatients. 1 in 30 children would be hospitalized, and 1 in about 650 children would die because of a rotavirus gastroenteritis. So based on this, we knew that we needed a vaccine, but we couldn't afford $200 a dose. And then the question was, given this history of oral polio vaccines needing multiple doses, would we have a vaccine that would work in India? So India and the US had an Indo-US vaccine action plan that had been there since 1987, and it had supported vaccine development of an Indian vaccine that was based on a neonatal strain that had been identified by Dr. Bhan, who was a pediatrician at the All India Institute of Medical Sciences. This was serendipitous. WHO had given him a kit. He needed to find stool samples. He thought the neonatal nursery was a good place to look. And then he found in children that were asymptomatic that there was a lot of rotavirus there. He then followed up those children and then looked to see whether they developed diarrhea subsequently or not, and showed that the children who had had a neonatal infection had fewer attacks of rotavirus gastroenteritis. So this served as the basis of thinking that this neonatal strain might be a potential vaccine candidate. If you had exposure to the strain that didn't cause disease, would it actually prevent disease? So this was taken under the Indo-US Vaccine Action Program. It came to the US. It was characterized here. Phase 1 studies were done in adults and in children. And in 1999, after RotaShield was withdrawn in the US, a company was identified in India to take vaccine development forward. And this was Bharat Biotech, a company that has grown substantially since then. But they were naive. They had never made a live viral vaccine before. It took them a really long time to get started, but they finally made the lots that allowed us to do clinical studies in India. By the time it got to be time to do the phase 3 studies that showed us whether the vaccine would work or not, there were already vaccines that were available in the Indian market. The Merck and GSK vaccines were available there. So then we had this question, could you do a placebo-controlled trial? So we had consultations with bioethicists around the world and finally with WHO, and had this recommendation that said that we could go ahead and do this trial, but we had to make sure that the kids that were in that trial were as safe as we could possibly make them. That meant that every family got a cell phone with the numbers of the study team, and they could call us for any problem their child had at any time. So essentially for 2.5 years, none of us slept. It was a big team, and all of us were on call all the time. And we wound up with a vaccine that had 55% efficacy. This vaccine was licensed by the Indian regulatory authorities in 2014, and this vaccine, which had been funded by the Gates Foundation, was under an agreement where it would cost only $1 a dose. So this made it something that our program could afford. Unlike the US, where the price of vaccines has gone up, in India, this vaccine for public programs now costs 60 cents a dose. Now, the big question was, I told you 95% efficacy of the Merck and GSK vaccine. So if you have a trial that shows you only 55% efficacy, that looks bad. But we had been doing work looking for a correlate of protection for rotavirus for a really long time. And in the work that we had done, we had shown that comparing cohorts across Mexico, India, and Africa, that protection from prior exposure to rotavirus was going to be incomplete. And we had predicted that the vaccine would have about 50% efficacy. Still good, but not as good as the vaccines in the West. But the one thing that I will point out to you is that this kind of study of natural infections gives you good information that comes from local regions. But the most important thing is what lower efficacy really means in developing countries. So here I'm showing you data not from the vaccine we worked on, but data from the GSK vaccine evaluated. Let's just take the example of Malawi and South Africa. In Malawi, the vaccine had 50%. The GSK vaccine had 50% efficacy. In South Africa, it had 77% efficacy. But if you look at the background rates of infection, in Malawi, if you followed 100 children for a year, 13 of them would develop severe rotavirus gastroenteritis. If you followed 100 South African children for a year, only 5 of them would develop a severe rotavirus gastroenteritis. So if the purpose of a vaccine is to reduce disease, with 50% efficacy in Malawi, because of the high rates of infection, actually you prevent about 7 episodes of severe rotavirus gastroenteritis, whereas in South Africa, you only prevent 4 episodes of severe rotavirus gastroenteritis, despite the fact that the calculated vaccine efficacy is much higher in South Africa. So we had shown our Indian Technical Advisory Group on Immunization that there was a significant disease burden. They wanted cost-effectiveness studies. They wanted WHO to recommend the vaccines. And finally, they wanted to know whether use of the vaccine would promote equity for the population. It's interesting that rotavirus vaccines were available, were recommended, were being used in the private sector from 2008 onwards. That the rotavirus vaccine was recommended for use for the entire population only in 2015. And in 2016, we were able to introduce the vaccine, initially starting at 9% of our birth cohort, but by 2019, taking that vaccine countrywide. India has now 25 million children born every year, and 90% of them receive the rotavirus vaccine. We also worked on a second Indian rotavirus vaccine. This was a product, again, made at the NIH, came in under the Indo-US Vaccine Action Program, was developed by the Serum Institute of India. This one was evaluated both in Africa and in India. It had higher efficacy in Niger than it did in India. This was a heat-stable vaccine, could be kept outside of the cold chain. And with these two vaccines, we finally had enough to vaccinate our entire country. So these were the first phase 3 studies that were done in India. And it's interesting that the two companies that made those vaccines have grown to become one, the most innovative company making vaccines in India, which is Bharat Biotech. They have the largest portfolio of vaccines, and Serum is the biggest producer of vaccines in the world. So it was a privilege to work with them and to work with international collaborators. All of us learned a lot through that process. And we also learned that, when there are setbacks, there are always people that you will find who will help you out. Just like to compare vaccine introduction in the UK and India, and I can take two examples here. If you look at the measles vaccine, that was introduced in the UK in 1968. It was introduced in India in 1986. And after that, India did not introduce a new vaccine until 2009, which is when we introduced the Haemophilus influenza type B vaccine as part of the pentavalent vaccine. Since then, we've introduced a number of vaccines, including the rotavirus and the pneumococcal vaccines. I'd just like to illustrate something to you. The gap, which was 20 years for vaccine introductions in rich and poor countries, has been changed over time because of one organization, and that's Gavi. It was founded in 2000 to address vaccine inequity, to make sure that kids in developing countries also got vaccines. But what I'd like to show you here, and I'd like you to do this exercise. You have the same color as a solid line and a dotted or a dashed line. So if we take a look, let's say, at PCV Gavi countries, you can see that the Gavi countries, the dotted lines, the dashed lines, always start later than the high-income countries, which are the solid lines, right? If you look at PCV and Rota vaccines, what you can see is that though we might have started later in developing countries, our coverage is actually higher than high-income countries. That was not the case for HPV vaccines. HPV, we started late, we are staying low. Why is that? Well, HPV isn't made by a developing country vaccine manufacturer as yet. Serum has just started to make HPV vaccines, and I hope that we will have the same pattern as we have had for the other vaccines happen for this as well. So I'd like you to now compare the upper map and the lower map. You can see that things have changed a lot. But our job isn't quite done yet. We still have a long way to go when it comes to preventing disease deaths due to diarrhea in Asia and in Africa. Now, just to leave you with this figure, in 2018, as was pointed out, the two Indian vaccines both became pre-qualified. And I was really, really pleased when last year's figures came out for Gavi's purchase of vaccines. And now 3/4 of Gavi's needs for vaccines are being met by Serum Institute of India and Bharat Biotech. So with that, I'd just like to say thank you to the institution that I worked in, the people that I've worked with, but mostly to the children that participated in our studies and their families.
Thank you, Dr. Kang, and thank all our amazing speakers this morning. I hope you're as inspired as I am. I think we've heard amazing science. We've heard stories of challenges, how to overcome challenges, and some of the challenges that still remain. So I don't have anything more to say except please enjoy the rest of the day. The laureates and the students who registered for the lunch, but only the students who registered for the lunch, please go outside, pick up a lunch and go upstairs, and the students will meet with the laureates over lunch. For the rest of you, please enjoy your afternoon, but we haven't finished yet. We have another important event here tomorrow morning, where we have an exciting global health symposium about pandemic preparedness, from vaccine technology to global health policy. And we have a stellar cast of international speakers, including, again, Dr. Kang. So please join us tomorrow, same time, 9 o'clock, same place, for the last event in Gairdner Science Week 2024. Thank you.